problem in Microsoft PowerPoint installed on my system. So I have to shift to another system. So today's topic we are discussing about is about drugs, which are very much essential for our health, for our well-being, but they have become a nightmare or a bane for the environment. So it is a catch-22 situation uh, where we cannot live without drugs. They are the essential nowadays, essential commodities. But at the same time, they are causing environmental pollution. So there has to be, so we will be discussing what are the various, what are the causes of these environmental pollutions by drugs, and to what extent these pollutions occur, and what are the various major drugs which are causing pollution, what are their effects on the environment, not just you see air, water, or so on, but the other animals or the other living organisms in the environment, they are being affected by these drugs, and how we can minimize, because we cannot eliminate this pollution, but we can definitely minimize this pollution. So drugs or medications, definitely they improve the quality of life as well as life expectancy. When we talk about the quality of life, like particularly in old age, see with the invention of more and more drugs, now more or more diseases are being treated, they have become treatable. As a result, the average lifespan of a person or a human being has increased. Now, as a person's, you see, lifespan increases, so there are other issues related to it. These are the you see diseases in the old age situation. Now, in old age, or we call it a geriatric state, there are some diseases which are very peculiar to it. For example, Alzheimer's disease, for example, Parkinson's disease. And these diseases, they severely affect the quality of life, because the person, the afflicted person, he cannot, you see, remember the things, or he is not stable, right? And he's not stable physically as well as mentally. So we are giving them medications, and those medications, they improve his quality of life. And as well as life expectancy, as we have already just mentioned. Now, from the Indian context, from the Indian pharmaceutical industry point of view, although this pharmaceutical pollution is not just centered to India, it is a global problem. But where India stands and why the pharmaceutical pollution in India is significant, or we can say it is underestimated, or it is you see under analyzed, and therefore not many people know about it. Now, if we see the Indian pharmaceutical industry, it ranks a third in the world as far as the production volume is concerned, and by value, it is at the 14th place. So now in India, there are more than 3,000 drug companies, and there are more than 10,500 manufacturing units. Now, these units, they are manufacturing drugs or products, and those products are being sold in India, as well as most of the companies, they are involved in the export business. Now, see here the export business. Now, India is the largest provider of the generic drugs globally, and it caters to more than 50% of the global demand of vaccines, 40% demand of generic drugs in US, and 25% of the generic demands in UK, right? And why this has happened? Because India has a very large pool of scientists, students, experts, And of course, government policies are there which are promoting industrialization. So that is leading to you see development of the Indian pharmaceutical sector. Another feather in the cap is more than 80% of the antiretroviral drugs which are used across the globe to combat AIDS, right? They are supplied by the Indian pharmaceutical industry. Now, this pharmaceutical industry has grown, you see, not in just a few years. It has taken several years, and we can divide this evolution of Indian pharmaceutical industry in four stages. The first stage we can say is before 1970, when most of the pharmaceutical industry in India that belonged to the foreign counterparts. Because at that time, Indian pharmaceutical industry has not evolved, it has not developed, machinery was not appropriate, so affordability was an issue. And then during 1970 to 1990, domestication of the pharmaceutical industry happened with the advent of Indian Patent Act. Just give me a few seconds. So stage two, sorry, there was someone outside. So there was a second stage is the 1970 to 1990 when the domestication of the pharmaceutical industry happened with the introduction of Indian Patent Act and the export initiatives. So with the help of government policies, their support, more and more, you see, industry, they get involved in the production houses. Then during 1990 to 2010, there was a liberalization of the Indian pharmaceutical industry, and that liberalization led to the ability of the Indian pharmaceutical companies to launch operations in the foreign countries. So in some Indian pharmaceutical companies, they become the multinational companies, so that helped them expand their business, and the world came to know more about the potential of the Indian pharmaceutical industry. The major growth happened, you see, during 2010 to till date. This was because of the shift of the, you see, community from ethicals to generics. The reason is the Western pharmaceutical industry, they were more into the ethicals, means the branded medicines, which are very expensive. And as the patent expires for a particular product or for a particular drug, it comes to the generic market, and the generic medicines, they are very cheap in comparison to the ethicals, in comparison to branded. Now, because Indian industry was not involved significantly into research of the new chemical entities or new drugs, so they were largely relying on the production of the generic drugs. And uh, because the now people, they or the, you see, the business community, they were exploiting the generic medicines more. So the Indian pharmaceutical industry, it has grown not only in India but also in the Western world also. Now, what are the various advantages which are posed to the Indian pharmaceutical industry in India that has resulted in the huge growth? That is a cost efficiency. Now, here we know that in India, the cost efficiency has, in comparison to the Western world, is very good. Means the labor is not expensive, the machinery is not expensive in India, right? So these has led to the, you see, reduced cost. Then there are several economic drivers, like now people are getting more concerned about the healthcare expenditures. They have started spending on their healthcare, on the medicines, right? They are not just relying on the traditional system of medicines, but they have, you see, now significantly shifted to the allopathic system of medicine. Then policy support, the government is also, infusing money to support pharmaceutical industry and increasing investments from the foreign companies or the foreign or the Western world. The reason of increased foreign investments is again the cheap or the less expensive labor and infrastructure here in India. If we see the clinical trials, so every drug which has to be launched in market has to undergo clinical trials. So India has become a hub of the clinical trials. Why? Because the patients who are recruited, they are given a certain amount of money, which is a part of the clinical trial protocol, and that money which is given to the patients in India, it is very less in comparison to the money which is given to the patients in the Western world. So it is a very lucrative option for the Western world to go for the clinical trials, to go for the products which are produced in the market. Now, Indian medical, not only in the Western world, but the India's medical expenditures that has increased significantly, and in the next 5 years, it is expected that it will increase by 9 to 12%. And if at this rate, it will make India one of the 10 largest nations in terms of medical spending. And compared to other nations, the cost of manufacturing pharmaceutical goods in India is lower and more effective, as we have already discussed. And this is, visible from the Sensex market. The BSE Healthcare, the Bombay Stock Exchange Healthcare Index, has risen with the annual growth rate of 12% per year over the last 10 years, and whereas a Nifty Pharma has given you a return of approximately 11% per year. So this is, a very significant return, or we can say this is one of the best returns of all the sectors which are, included in the Sensex. Now, what are the various types of drugs which constitute the Indian pharmaceutical industry market? See, the 13% of the drugs, they are the cardiovascular, 16 are anti-infective or antibiotics, 11% gastrointestinal, then vitamins, respiratory, analgesic, and 29 are others. Others, they include the miscellaneous one. See here, the main components, they are the anti-infective and cardiovascular and gastrointestinal, and these are nothing but lifestyle related problems. So what is this, how a drug, undergo different stages during its life. So from the manufacturer, it goes to the distributor, from the distributor to physicians to hospitals and to patients, and then to the wastewater treatment facility, means after the user, it is excreted in the environment. Now, this is the fate of pharmaceutical products in a broader way. See, the households or the non-domestic points like industries, hospitals, or other services where the drugs are manufactured or stored or used or dispensed. So these, initiators, the households or the non-domestic points, they release the drugs into the environment through septic tanks, through direct discharge, means by the industry, effluents discharge. Then they are discharged into the sewer. Now the sewer, it goes to the waste water treatment plant, and whereas the direct discharge, which is let's say some effluents that they are directly discharged into rivers, freshwater streams, right? They mix with the surface water. Then the septic tanks, which are made in homes, right? From there, the medicines or the drugs, they are diffused into soils, right? Then we have animal farming also. Animals, they are also given drugs to increase their milk production, to increase their body mass. So those drugs, they include, steroids, those includes birth control or the reproductive drugs, or so on. And these animal farming, they excrete and their excreta is used as manure in the solids in the farms, and so they are spread over the soil. So from these biosolids or the manures, right, and these biosolids, they are also formed from the wastewater treatment plant. So what is done in the wastewater treatment plant? The effluent, it is treated, and it is split into two parts. One is the treated effluent, or we can say treated water, and another is a sludge. So that sludge and the excreta from the animal, they are no doubt an excellent source of manure, but at the same time they have drugs remained in those, because wastewater treatment plants, they cannot remove the drugs completely. Their efficiency is just 30 to 70%. Rest of the drugs, the rest of the component, they remain in either the effluent or in the sludge. So that sludge or that excreta, or that is spread on the soil. So soil is now rich in the drugs or the medicines. And this effluent is then treated with is mixed with the surface water. And now we have the aquaculture also, where you see fishes or other water animals, they are grown from the commercial point of view. And that also, not sea, the freshwater animals, aquatic animals, they are again need drugs to maintain their growth, to maintain the asepticity in those cultures. So surface water is contaminated, then soil is contaminated, right? And from the soil, right, the drugs, they leach into the groundwater. Now, this seems to be some improbable situation, but around 30 years back, a study was conducted in India, where it was found that a hand pump which was installed near to a dairy farm, that groundwater was found rich in diclofenac. Now, from where the diclofenac has, come into that groundwater, it was because the animals, they were given diclofenac, and the diclofenac, it is excreted as such into the urine. And see, the so many animals, they are excreting urine, right? And that urine is getting dumped onto the soil. So that urine, followed by the rain, followed by water, it gets slowly leached into the groundwater through some channels, right? So this is the, these things do happen, right? The leaching of drugs from soil to groundwater. And then from groundwater or the surface water, we get drinkable water. And drinking water again fits the household. So this is nothing but what we are excreting, we are getting it back, right? So this is way the pharmaceutical drugs, they are contaminating the whole environment. They're contaminating the soil, they are contaminating the water, right? And they are also contaminating air because of the pharmaceutical industry. So there are several reports nowadays which say that 613 pharmaceuticals are reported in the environment worldwide, and there may be more, but because there may be more because so they need more sophisticated or sensitive analytical methods. Now, even the rivers in all regions of the world, they are contaminated with many medicines. Uh, so in 2007, there was a shocking finding in India. There is a wastewater treatment plant near Hyderabad, and that wastewater treatment plant is catering to some around 100 pharmaceutical industries in that area. So the affluent of those around 100 pharmaceutical industries go to that wastewater treatment plant, and that wastewater treatment plant treats the water effluent and converts into sludge and the effluent, and that is the treated water, and that treated water is used for further drinkable water plants or for the farming and all that. Now, in that effluent, test was carried out, analysis was carried out, and it was found that it is highly rich in antibiotics. Highly rich in antibiotics. The concentration of antibiotics in that effluent was found to be even greater than the maximum permissible antibiotic concentration in blood of infected person. So this is the, you see, extent of contamination of our waters, right? And wastewater treatment plants cannot remove those antibiotics or those chemicals to significantly. Their efficiency is just 30 to 70% if they work with full efficiency. So that those antibiotics they are released into what? Effluents, and they mix with the fresh water, and fresh water we are again taking. So that is leading to antibiotic resistance. That therefore you see in the in the previous slide, we are seeing that antibiotics, they are constituting 16% of the market. Why? Because we all are getting resistant to you see antibiotics. The bacteria, they are getting resistant to antibiotics. Therefore, we are needing a stronger and stronger antibiotics. So antibiotic market is, you see, it is bound to grow. It will further grow because this menace of mixing of these drugs with the fresh waters, it is not going to stop very soon. So regions, rivers in all over the world. This is not just India. This in case of India, it is all over the world. They are contaminated with many medicines. There is another study in Michigan Lake. In Michigan, there is a lake, and that lake has been found to be contaminated with metformin. Now, metformin is a potent anti-diabetic drug, and concentration is significantly high. And just imagine the region up to which the metformin was found. It was up to 5 kilometers from the shore. So that is a huge lake, and there is that is contaminated with metformin. So what is happening to the aquatic life present in those you see river water bodies? Similarly, in England, you see anti-inflammatory drugs and painkillers like ibuprofen and diclofenac, they have been found in fur taken from otters. In Sweden, right, the perch fish, they were found to be contaminated with 23 pharmaceuticals, and these fish, they are used as you see food in those countries, even in India. And those pharmaceuticals, they're antidepressants, sedatives, antibiotics, painkillers, or anti-cancer drugs. This is a very major issue, issue of concern. Anti-cancer drugs, they are highly cytotoxic. Then Baltic Sea, in Baltic Sea, there are salmon, they are found contaminated with ethinyl estradiol, which is used in the contraceptive pills. Nowadays, the uses of contraceptive pills among humans is very rampant. So that is again another source of contamination to the environment. Now, dozens of medicines are found in samples of drinking water in European Union countries with large monitoring programs, for example, in UK, France, Sweden, Spain, Germany. So they all actually in these countries, they don't have very, you see, fresh source of waters. So they are getting the drinking water either treated from the sea water or from the effluents, and they are again, that's why their drinking water is getting contaminated with more medicines or more drugs. So these drugs which are found in water, right, they have shown a very harmful effect on the laboratory animals also, because some experiments are carried out at the laboratory scale, like some experiments are carried on fishes, on otters, on insects, or several species, right? Their laboratory animals, they have shown severe effects by these medicines. Despite this, there is a little monitoring of the, you see, discharge of effluents into the bodies. There is a little, you see, monitoring of, you see, disposal of drugs. Therefore, there is no yet legal standard for the residues of medicines in rivers and drinking waters. And this all has happened, right, so rampantly because till year 2005, there were no standards, there were no protocols for the assessment of environmental risk from these drugs. So after 2005, there were some standards set, there were some protocols set for the assessment of environmental risk of human medicines. Then it came to fore. Yes, this is the extent of this contamination. Now, because wildlife is decreasing due to this environmental contamination, that environmental contamination may not be due to the drugs alone, but there are several reasons why the wildlife is declining. And out of this, you see 31% is due to habitat degradation or change, right? 7% climate change, 37% exploitation. But here, habitat degradation or habitat change is amounting, is a compound, you see, contributing 31% for the reasons for wildlife decline. Why we are seeing habitat degradation? Habitat, when we are discharging drugs or pharmaceuticals or personal care products or any chemicals into rivers or into water or in air, right? That those drugs, they are coming back into the soil and they are basically changing the nature of that habitat. We can say the habitat has degraded or the habitat has changed. So therefore, it is causing the wildlife declining. Now, what are the various sources of pharmaceutical pollutions? One is the main is the effluent, right? Then there is a disposal of the unused or expired drugs and products. Now, uh, not only in India, all over the world, this is a general practice that we all have medicines stored in our homes, and some of the medicines, when the treatment is over, they either remain unused or they some of the medicines they get expired at our home. How do we dispose those medicines? In 99.99% of the households, those drugs are either thrown into into the dustbin or they are just flushed, right? And what is happening to those you see drugs which are discarded in the flushes or in the dustbins? They get mixed with the garbage, right? And that garbage is again used as a manure, and that manure we are feeding to the farms. So this is again a recycling. So whatever we are throwing, we are getting back. Then human and animal excretion of drugs, we have seen our body excretes around the 90% of the drugs unaltered. Although our body metabolizes it, but around the 90% of the drugs they undergo unmetabolized. Then there are healthcare institutions like hospitals, nursing homes, where the drugs are used in a huge volumes, and they have discharges. Then drug manufacturing units, drug manufacturing, there's a major thing. Then agriculture. Now, agriculture is also contributing to the pharmaceutical pollution because the animal waste which we are obtaining from the poultry farms or from the cattle or other livestock, they are again mixed with the drugs because they are given reproductive drugs, they are given antibiotics, they are given painkillers for maintaining their health. And those animal waste, they are used as a manure, and again through agriculture we are promoting the pharmaceutical pollution. Now, these pharmaceutical waste, they contain, they're not just drugs, but they also contain heavy metals. That also includes dust, which is generated during formulation. Then these are the gaseous vapors or emissions during the synthesis of the drugs in the industries. And there are some chemical spills also. And these pharmaceutical waste, they are highly ecotoxic, carcinogenic, they are persistent, and therefore they are bioaccumulative, resulting in a compounding negative effect on environment and human health, and as well as animal health. So like water pollution, right? So this is because of the spillage, drains, and generation of effluent water, sewage water for uncontrolled pH, air pollution, land and soil pollution, noise pollution is there, noise pollution that also leads to, you see, disappearance of, or the extinction of certain species. Now, in United States, Spain, Germany, and the United Kingdom alone, there are over 101 different pharmaceuticals which are present in groundwater, surface water, drinking water, or tap water. So situation is all over the world, in Thailand, Canada, Australia, India, China, South Korea, Japan, everywhere, right? There are around 100 different pharmaceuticals present in waters. So these are the some documented studies on the treated industrial effluents. See the term treated industrial effluents. So after the treatment, whatever we get, those are tested for this drugs, and see it's in all over, every country. In India is here also, salicylic acid, right? Many fluoroquinolone antibiotics, and see the concentration 2.7 grams per liter, significant amount. Ciprofloxacin 31 milligram per liter. Its a normal dose is 250 milligram or 500 milligram depending upon the disease, right? And in 1 liter of that effluent there is a 31 milligram. Then again in India, see ciprofloxacin, cetirizine, again see here the India fluoroquinolone, ciprofloxacin. So they were these analyses were carried out at the different time periods from the different locations. See ciprofloxacin also found in the soil, right, or in the organic matters, 7.2 microgram per gram of that. So situation is not very good as far as the contamination is concerned. So many studies or many reports are being reported by the different scientists from across the globe. Like more than a quarter of the world's rivers, they contain potentially toxic levels of over-the-counter and prescription drugs, including antihistamines, antibiotic, caffeine. This is a very recent report, 14 February 22. Then there is another 16 February 22 report. Drugs are polluting our rivers, threatening humanity and environment. So awareness, people they are getting aware, but again the more actions are to be required at the regulatory level, as well as what we as individuals can do to prevent this pollution. We should do our bit. So this is another study in which across 104 different countries, 258 rivers were studied, and they have been found in across the different subcontinents, and they were found with many drugs. And what is that? The higher concentration of the pharmaceuticals areas that had a higher median age of residence. Of course, because as we become older, we need more medicines, and therefore those medicines are discharged into the waste, and they are getting reflected in rivers. So out of those 104 countries, 258 rivers, right, there were around 61 active pharmaceutical ingredients. And the most frequently detected ingredient was carbamazepine, which is an anti-epileptic drug, metformin, anti-diabetic drug, and caffeine. So these are the three drugs which were found in almost at least half of the sites which were tested. Means out of 258, around 150 they were found. Then antibiotics were also found at the dangerous levels in around 20% of the sites, and those antibiotics are in such concentration that they are harmful to the wildlife, to the aquatic life, or so on. And the major hotspots which were detected during this study, they are the Lahore in Pakistan, La Paz in Bolivia, Addis Ababa in Ethiopia. Now in Spain, from where our international faculty is from, the Madrid is a city, and it is around 10% of top 10% of the places with the highest cumulative concentration of antibiotics. Glasgow, Dallas. So there are the other cities also which are highly affected by these drugs. So why the rivers are most affected? Why the rivers are studied? Because rivers are the bottom of the system. of an ecosystem. Rivers are the bottom. Whatever happens on the land, it all ends up in the river, right? Whatever we excrete, it all goes to the fresh water streams, right? So in Ganges, see here, the tens of billions of effluent are dumped in Ganges in India every year. So because the pharmaceutical industry or for the chemical industry or agriculture industry or metal industry, so all industries, they are lying, you see, closer to Ganges, and their effluent is being flushed into this Ganges, polluting the Ganges. See here, this is another example of a river in Hong Kong where 34 different drugs were detected. Now, this is, you see, a regional distribution of various drugs. Now, these countries, they are belonging to the different regions as per the United Nations. Like this is the Western Europe and other group. This is the Eastern Europe group. This is the, you see, Latin American and Caribbean. This is the Asia and then Africa. And see here the different kind of drugs which are found. So this is the percent of the MEC database entries, means how many, what is the concentration of, what is it, this is indicating the extent of prevalence of these drugs. See here the antibiotics, the maximum in Asia, maximum, right? What can be the reason? Because in Western world, like Europe and American countries, they are their antibiotic sale is very much you see over the counter. So in India particularly, let's say in our neighboring countries also, if we see that these regulations are not strict. If we are having some infection, we can directly buy antibiotics from any chemist shop without any prescription, right? So that is a rampant use of antibiotics in Asian region. But this kind of sale is not, you see, allowed in the Western world. Now in Africa, this again, you see, the antibiotics are less used. That may be because of their inherent good immunity towards those microbes because of it being a temperate region. Then see estrogens. Estrogens are used maximally in Africa, then here the Latin American or Caribbean states. So this is the worldwide distribution of this different drugs across different countries. So every country, every region is rich in one or the other form of drugs. And if we divide the different regions, and then if we do the comparative analysis, right, there are 646 drugs were tested, and out of those 574 were detected in the Western European group. And these are the some common things which are observed in different countries. So these are the various drugs which are mainly detected. So there are the some top 10 to 15 drugs: diclofenac, carbamazepine, ibuprofen, sulfamethoxazole, antibiotic, naproxen, analgesic, and then estradiol. They are estrogens or the regulatory reproductive drugs, right? Then paracetamol, trimethoprim, antibiotic. So mainly these are the estrogens. They are the antibiotics and analgesics, which are mainly excreted, which are mainly contaminating the environment across the world. So this is the, you see, a pictorial representation of, you see, the number of pharmaceuticals detected in surface water. This is the surface water across different worlds. This is the maximum one, and this is the, yeah, this one is the minimum one, right? And then this is the in drinking water. This is the surface water, and this is the drinking water, right? Now here, drinking water is a maximum in, you see here the European country here, Portugal is there, Spain is there, and here is some near to Netherlands. And India, there is not mentioned. So but there is nothing to be happy about, because the data is not available. in India, from the Indian context point of view, if we see the reports on testing of drugs in drinking water, you will rarely find any report on this because the analysis or the studies are not being taken up. Then this is you see diclofenac specifically. Diclofenac is maximum here in Brazil, then in European countries, right? Then in Pakistan here. Then here it is diclofenac is the maximum again here, right? For diclofenac, this is for the ethinyl estradiol, the estrone, maximum in American countries, right? Also China, right? So there, what can be the reason specific for that? That may be attributed to their reproductive problems or increased use of contraceptives. So these are the various causative factors we have already discussed, and these fresh waters, they are affecting the ecosystem very grossly. In US, this is a European eel in this Europe, and this is a hellbender salamander in the US. Their population was significantly reduced. Since 1970, it has reduced to 75%. So their growth has been affected. But now the governments, they are getting more and more cautious about it. They are generating policies for the effluent treatment, for the release of the effluents into rivers, and therefore the recoveries are now being seen. Similarly, in England, there is otters. They were near extinct, but now they are being conserved by other means also. So their extinction, or they are near extinct because the drugs which we are using, they are contaminating their habitat, they are degrading their habitat. Vultures, which were very commonly seen in India before in 1980s or in 1990s, early 1990s. They were very common. But those vultures are nowadays almost extinct in India. Almost extinct. They are not seen. Why? This is because these vultures, they feed on the animal carcass. The animals, the cattle which are dead, they are dumped at specific sites, and from those animals, these vultures, they feed. And these animals which are dying, which are dead, they are dead because of certain diseases, and during their treatment they are given high amount of antibiotics and also diclofenac, right, to relieve the pain. Now, it is this diclofenac responsible, which is accumulated in their body, and those diclofenac is eaten by this vulture, and that diclofenac is lethal to this vulture. Therefore, the vulture, they have almost now disappeared. This is the, you see, direct effect of these drugs on our ecosystem or our environment. Like anti-parasites, like ivermectin, which is a veterinary drug, imamectin, which is a fish farming anti-parasite. They are excreted in the feces of the treated animals and adversely affect the invertebrate organisms that live or feed on the drug. So these drugs, they are affecting the farm animals as well as the aquatic animals. Antibiotics, we have already seen that antibiotics, they are causing resistance. The major issue is the resistance. It is estimated that 5 million people died in 2019 alone because of the antibiotic resistance. This is a major concern, and according to the experts, the antibiotic resistance is going to be the next pandemic, because the antibiotic, even in our daily life, what we do, if a doctor has prescribed us a medication for, let's say, a week, antibiotic for a week, and we feel better after, let's say, 4 days, we don't take that antibiotic for the other 3 days. So what is happening in those 3 days? The bacteria which are they get just inactivated, they are not killed. And when they are inactivated, it means some mutations, they happen in those bacteria or on those microbes, and they become resistant to antibiotics. So next time when we get again sick, right, and we get if we take that antibiotic again, we have to take the increased dose of antibiotics, or we have to change the antibiotic, right? So this is the, you see, short for this is the fallout of antibiotic resistance. Now, a study in that Hyderabad, which was carried out in 2007, there were antibiotic resistance, and their antibiotics were found in the effluent, and there were some bacteria which were found in those rivers, in those systems, and those bacteria were found resistant to the most potent antibiotic available at that point of time. It means if that bacteria somehow infect any person, that person could not be treated. So this is the, you see, high time we have to think about antibiotic resistance. We have to be rational about antibiotics. Ibuprofen, that causes a male fish to abnormally take the female egg yolk protein. See here, they are changing the physiology altogether, right? So that is called feminizing of male fish. So male fish is becoming a female fish, right? Or we can say the dual sex fish or dual sex organs, right? This is, you see, a major bolt to the population of fishes, to the aquatic population, resulting in, you see, disappearance of certain species. Ethinyl estradiol, they are, you see, they are used in contraceptive pills, and they are also used to treat menopause. And when this estradiol is consumed by male fish, they are feminized again. So ibuprofen, ethinyl estradiol, they are causing feminization of the male fish. See, the synthetic estrogen, they are used in birth control, same one, right? So there was a one study which was carried out in Ontario, a lake in Ontario, in which this fish, which is fathead minnows, they are the small fish ranging from 7 to 10 centimeter, right? And their population was severely affected. Severely affected. See here, their fathead minnows, their population decreased by almost 99%, means almost complete, right? But at the same time, there were certain species which were increased. For example, here, Cabora species, right? Rotifers, they decreased, right? Littoral, they increased. So there are insects which increase. Why these insects increased? Because of pollution. Because these fathead minnows, they feed on these insects. And if these insects, if these fathead minnows are not available, so they will not be eaten up. So their population will increase. So population of insects increase, population of fish decrease. So you see, there is a total ecosystem. One drug is affecting one aquatic animal directly, and it is affecting the other animals indirectly. So if there are too many insects, they are causing another health problems, another health hazards. See here, this is their results, which have shown that this is the insect species which has increased, population is increased, littoral insects, they're increased. Minnows, this one, the fishes, their population decreased almost to zero, and they are also fathead minnows in the young age, and the adults both decreased because of those drugs, ethinyl estradiol. Now, another type of drugs which which are very much detrimental to our environment, these are the addictive drugs. The drugs which are used for the recreational purposes, which are used for, you see, for getting a high, right? Like marijuana or cannabis. This is industry which is very much, you see, which has grown significantly in last 20 to 30 years. And because its demand is very high, particularly in the Western world. And see, and this cannabis, it is grown in the greenhouses to increase its production, to increase its demand, meet its demand. It is grown in the greenhouses. And what is the implication of this cannabis production? Now, we are discussing it in a different heads. One is the carbon footprint. All right, what do you mean by carbon footprint? Now, cannabis production In the US alone, it accounts for around 1% of the country's total energy consumption. Total energy consumption is high, very high. Within a single year, more than 15 million metric tons of carbon dioxide are emitted in the US as a result of the indoor cannabis production. And this 15 million metric tons of carbon dioxide is equivalent to the annual emission of 3 million cars. See, so that is the extent of, you see, environmental contamination or environmental pollution by cannabis production. It means if we are taking a single joint of cannabis, right, one shot of cannabis, that is equivalent to 3 kilograms of potatoes. Carbon footprints of means carbon dioxide generation by 3 kilograms of potatoes. So that is the extent of, you see, energy consumption by cannabis production. Then another is a water stress. Marijuana is an extremely thirsty plant, like paddy in this region. Paddy is another very thirsty plant where it needs, we know that it needs a huge amount of volume of water. Similarly, cannabis or marijuana, they also need a huge amount of water. And this cocaine or the, you see, cannabis, it has a fuse deforestation. Deforestation. Why deforestation? Because cannabis, it is although it is it's a narcotic drug, but we get license to grow it, and some forests, they are the government. It is a government license, right? We don't get license. It is produced only under the you see surveillance, under the control of government. So government. They have converted their forests into the cultivable lands for cocaine. So as a result, the forests they are decreasing. Now, Colombia, South America is the you see major hub for this cultivation of cannabis, where 741 million acres of forest is used or it's cleared in South America alone. And out of this, in Colombia only, right, 50,000 acres of Amazon region alone is converted into cannabis into land for cleared for the cannabis production. And out of this 50,000, around 50,000 acres, right, 50,000 hectares of this land is a nature reserve that is a home to a rich diversity of species. So those diversity of those species, they will ultimately eliminated or they will die in those land because it is now being used for cannabis. Now, coca leaves or the cocaine, or you see, cocaine is a product of coca leaves, and cannabis or coca leaves. They are not just grown in jungles; they are also processed into cocaine. So, cocaine is an end product of coca, right? And cocaine, to convert it into cocaine, it needs several chemicals, toxic chemicals like ammonia, acetone, hydrochloric acid, or so on. And these, because this conversion from coca to cocaine is illegal, so this you see conversion is done in a very clandestine way, using some illegal means. And so the whatever the waste is generated, so there cannot be you see a regulated way of releasing of those waste into the wastewater treatment plants or so on. So they are either flushed into the freshwater streams, or they are just dumped on the farms, or they are just found on the roads. So this picture which you are seeing is provided by the Belgian police, where they have found that some industrial waste from the cocaine, they were you see dumped on the roadside near a body of water. So this is causing what pollution of you see our system. And this waste is sometimes dumped into containers used to collect cattle excrement, right? The cattle excreta, it is collected in those. So they are mixed with the cattle excreta, and they are, you see, spread on the crops. And that crop is found to contain amphetamine and some ecstasy residues. from where this amphetamine is a CNS drug, right? From where these are coming? They are coming from these, residues, these leftovers. Poppy is, another plant, another drug, addictive drug, which is again causing a huge environmental loss. So this I have covered because it is somehow related to the pharmaceuticals, because these are the drugs. So when we say drugs, not just the pharmaceutical medicines, but the drugs also which are used in an illegal way, right? So if we are excreting the drugs, we are contributing towards its pollution. And if we are promoting the, cultivation of some specific type of plants, it means that is also contributing to pollution, right? So this poppy plantation, it is again a very thirsty plant, and it is of a region in this Afghanistan. Now in Afghanistan, the groundwater is sinking by 3 meters per year, and because the land is good and so conducible to poppy plantation, but the only problem is the water. So water is available on the underground, and so they put solar water pumps to, take out water, and that water is used for this plantation. So more and more people are shifting into this region, and more water is getting pumped out of the ground. And the people or the scientists, they are fearing that after a few years, it is a possible that no plantation will be possible.
हाँ.
Seema ji, please mute yourself. Seema ji, please mute yourself. Thank you. So the local population or the scientists, they are fearing that after a few years, that particular area of Afghanistan may not be habitable because there will be no water. So if there is no water, no one can survive there. So there will be no crop, no plantation, no human beings. So what are the different ways to reduce this pharmaceutical pollution? There are few. One is limit bulk purchases. We have to limit the use of medicines. Only the medicine should be taken when it is of a dire necessity. This is what we can do as individual. This is what we can contribute to towards the environment, right? So let's purchase drugs as less as possible. Then there should be some some drug take back programs. Now this drug take back programs, they are quite effective for the pharmacies. Like if you go to some chemist shop, so there is a provision that whatever the medicine expired, the pharmaceutical industry takes back that pharmaceutical expired drugs, and they either recycle it or they dispose it in a different ways, right? So they have a standard set procedure. They incinerate the drugs. When their drugs are incinerated, they are converted to ash. So it is a converted to carbon, hydrogen, nitrogen, oxygen, and sulfur, because this is what a drug is composed of. So they are just converted to organic waste, which is not harmful. But as far as the local households are concerned, what we do, we simply flush the drugs. We flush the drugs in nearby streams, or we just throw them in the dustbins, right? So we have to avoid this. We have to stop that practice. So what we can do, that if there is some expired medicine, we can hand over that expired medicine to the chemist, right? So that it can be recycled, it can be taken care of. Then we have to be again careful about how to throw medicines into trash. That is also important, how we throw it in trash. If we are throwing it in as such, that is again harmful. So let's say if we have few drugs, so it's better we burn them at home, and after it is burned, it is converted to ash. That ash should be thrown into trash. Now, this is at a small scale. On a larger scale, what we can do to minimize this pharmaceutical pollution, for that, the Environmental Protection Agency of US, it has taken a four-pronged approach. One is the public education. We as a human being, we as a public, as a society, we need to be, be aware of this extent of pharmaceutical pollution, its implications, and how we can, prevent this pharmaceutical pollution. So we have to be educated first. Second is stepped up monitoring of water supply. So whatever the water is being, provided to any community, it has to be tested. So that is at the government level that we have to, that the government has to do, or regulatory agencies have to do. Then there should be partnerships with the healthcare facilities and agribusiness to reduce waste. So they have to be sensitized. They have to be, given the new technologies so that they can reduce the waste, and they can recycle whatever is effluent, whatever is generated. And probably new regulations. New regulations are required. And as an introductory step, this Environmental Protection Agency, it has added 10 pharmaceutical compounds that include 1 antibiotic and 9 hormones to its watch list of potentially harmful contaminants that warrant greater investigation. So they have mentioned that these are the 10 compounds, 1 antibiotic and 9 hormones. They said that any drinking water has to be tested for this. If it is found, then some actions have to be taken so that its a source can be ascertained and it can be prevented. Thank you so much for your attention. So if you have any query, please feel free or raise your hand. Is there no query?
Hello, Dr. Bansal.
Yes.
Good morning.
Good morning, Ji.
Sir, it was a nice presentation.
Thank you.
And I have a query.
Yes, please.
Even though there are strict regulation on the biomedical waste produced from the laboratories and the hospitals, etc., even though there are a chance of leaching or spreading these contaminants, medical contaminants from these sources to the environment.
Yes, there are, biomedical waste. When it comes to biomedical waste, there are strict regulations are there. And these hospitals, they definitely, dispose of those biomedical waste. Even the laboratories, they dispose of the biomedical waste. But when it comes to biomedical waste, usually that biomedical waste includes, if we see the hospitals, right, that hospital biomedical waste include generally the tissues which are removed during surgery, some mask, or some materials which are used during procedures, right? And definitely but they are not including drugs. When it comes to drugs, they have either the take back programs with the pharmaceutical industries, right? But because lot many patients, they are admitted into hospitals, and what about the excreta of those hospitals, of those patients, right? So that is again, that goes unnoticed.
Okay.
If there is a microbial waste, right, like we are working in a biotechnology laboratory, right? So we are some biomedical waste includes bacteria, the cultures, the medium which are used for the growth of bacteria, or the fermentation media which are used. So those are you see disposed of as a biomedical waste. What about the drugs? That's the point. See,
Okay.
if you go somewhere in any if you go to any pharmaceutical industry or any chemical industry or any metal industry which is you see discharging its effluents into nearby streams, so as per regulation every industry has to has a treatment plant. Only after treatment
Right.
it can you see discharge its effluent into the nearby stream. And if you go to collect a sample, right, before the treatment and after the treatment,
Right.
you will not be allowed to take sample after the treatment.
Sir,
Then
there is collection of lot of biological fluids like urine samples, blood samples. So what about them? What happened to them?
where in case of in that's what I'm saying, in the clinical laboratories you're talking about.
Okay, they are burned.
No, in clinical laboratories, see, again, in the clinical laboratories, the diagnostic laboratories, right? They are taking our
Right.
blood samples, they are taking our urine, they are taking our feces, right? So they are
Right.
testing it. What they are doing after that? They are not disposing it to some biomedical waste van. They are just throwing into the sink.
Yeah, that's why that's what I am asking. What happened to that those samples which contains a lot of metabolites as well as pure drugs,
They are
drug
generally
ingredients?
flushed. They are generally flushed. They are generally thrown in the sink.
Yeah,
Only some very, you
that's
see,
what they are getting into the environment.
obviously everything which is flushed into the you see sink, right, that will mix with the sewage.
Yeah,
That sewage will go to the sewage treatment plant,
certainly.
right, and that sewage plant has not a capacity to separate or to you see neutralize those compounds,
Right.
because those plants they simply work on the principles of separating the solids, right? Then there is some with the help of a bacteria, they degrade it, and then they are oxidized with the help of UV or ozone, and then it is a leftover. Now, if they are using ozone, now ozone is a very expensive technique. Now, if they're using ozone, right, then most of the pharmaceuticals can be, most of the chemicals can be degraded, right? But usually the usual oxidation processes are not too effective to degrade those substances. So they will ultimately go to the freshwater streams, and as a sludge, they will mix with the manure and go to the farms.
So it means these diagnostic labs should be prohibited to throw away these kind of samples right into the sewage or in the
Definitely,
wash basin.
definitely. See, the major diagnostic laboratories, which are so-called branded laboratories like Lalpath or SRL, or so
SRL.
on, they may have some mechanism, right? They may
Okay.
have, because you see they follow strict rules, right? But I'm not sure whether they follow it. No, but this is what I think, they may follow it, right? But about if we see the local laboratories, right, I haven't seen any you see van standing in front of their gate to collect their waste.
Yeah. Yes, we yes. Mhm.
So that is the
Right.
situation.
Thank you, sir.
Yeah, so
Thank you, sir.
that is from their point of view. But what we as a person can do, at least we can buy the medicines as less as required. And if there is any leftover, let's don't throw it in trash. Don't flush it in sink, right? That is what we can do from our end.
Okay. Thank you, sir. Thank you very
Thank you.
much.
Thank you. So any other query from any other participant?
Yes, sir. May I ask?
Sure, ma'am, please.
Good morning, sir. I am Dr. Renuka Das from Srimati Kishoritai Boyer College of Pharmacy, Kamptee, Nagpur.
Yes, ma'am.
First of all, thank you so much for such an informative session. And sir, I think that with the increasing awareness about all such issues, the Western countries have already started working towards the sustainable restoration of the natural resources.
Hmm.
And how can we, as a pharmaceutical education institute, contribute towards ensuring avoiding such pollution? Like apart from making the students aware about it, what else can we do? Could you please suggest, sir?
See, uh, again, as a pharmaceutical educationist, first thing is that this part of pharmacy uh, is rarely, I think it's not taught in our curriculum. It's not a part of our curriculum. I think in none of the subject this topic is covered. So first thing, it has to be, made subject, not a complete subject. At least it should be a part of our education, pharmaceutical education. What is the pharmaceutical pollution? How it can be? Although environmental education has been made compulsory in India, particularly in Punjab, I don't know the other regions of India, but environmental education is made compulsory, but that environmental education, if we see the content of that, it is mainly talking about air pollution, noise pollution, different types of pollutions, how they are affecting it, right? But as a pharmacist, what I think that we should make this pharmaceutical pollution as a component of our pharmaceutical education, so that the pharmacists which we are producing, they should know the effects. So when these pharmacists, they come into market, in the community, into the pharmaceutical industry, they should know what is its implication, what are the rules, and how it should be done. Then second part is, of course, pharmaceutical industry. Now this, to guide pharmaceutical industry for the proper way discharge of this waste, there are set of rules. As I said, there are rules are there. Like every pharmaceutical unit or every industrial unit, they have to have this treatment plant, right? But who is following, right? So as a pharmacist or as a educationist, right, we can educate our products means our students, right? And when they go to the industry, let's see how much they can contribute, right? And we have, drug regulators. Drug regulators are, most of those regulators are either our contemporaries or our students, or some are our teachers also. So that kind of awareness has to be, carried out, right? So that they take strict actions about this. This is what I think we should do.
Yes, sir. Thank you so much. As rightfully pointed out by you, that the environmental sciences subject, it is very much there, but then the content is what the students already study in their 11th, 12th standards.
Yeah.
So I think that according to pharmacy or the drugs, we should include the deleterious effects of these drugs or harmful effects on the environment. So thank you so much, sir.
Yeah, this is one thing, and we also have to include how these drugs are polluting. That is also important.
Yes, sir. Yes.
At least in every household where a pharmacist is there, right? At least in that household, there should be some safe disposal of drugs, right?
Yes.
And
Yes.
that pharmacist or that household, if that household motivates 2 or 3 houses in their neighborhood, right, to do that, right, then that will be a very big contribution from our side, right, at an individual level.
Yes, sir. Thank you so much, sir.
Thank you.
Hello.
Yes, Dr. Mechi.
Sir, good morning.
Very good morning.
Actually, I was listening that question answer session and your talk also. Uh, right now you are just talking about the household. As an individual also, or if I'm talking about my kids, we are having so many medicine and we are dumping in that our regular garbage and all. So what is a safe use? Because so many medicines or like those liquids or those tablets, they are after expiry or before in those medical stores, they are purchasing medicines and they are dumping in those garbage. So is it affecting that soil microflora or other environmental pollutants? Like even industries also, they are just making antibiotics and all, and it is going to pollute the rivers also. So as an individual, where we can contribute? I'm not talking about as a academics or as a research person. As an individual, if I have to create awareness to my neighborhood or whatever that person who is working at my home, so how I can do it that as a layman, because that is very important.
Yes. Uh, Actually, this see, the response to this query is in continuation with to the previous Dr. Renuka Das. See, as a layman or as a normal person, right? What we can do is that we can derive a mechanism very small at individual level, that we decide that we will not dispose of the drugs in garbage, right? So what we do, just take a small box, whatever the medicine is unused, whatever the medicine is expired, put into that. Label it as unused, unusable medicines or unused medicines. Sensitize your neighbors. If they are not doing it, ask them, okay, I have this box. If you have any medicine unused or expired, put into this. If we collect it, if they are not doing at their level, but at least they can give it to you, right? See, the kids, they are very much sensitive to these issues nowadays. The children who are studying in schools, they are very much sensitive. We can sensitize them कि बेटा ये ड्रग ऐसे नहीं करना, इसको यहाँ डालना. Take, if you have any friend, if he has some medicines which are not usable, ask him to bring it and put into this box. Right? After collecting it, let's say from 10 or 5 households, right? Then those medicines can be given to a chemist or who's responsible. We know that we have some chemists everywhere, right? And some of the chemists are the friends or all, they are known to us, right? Although every chemist will not take back those expired medicines because that increases their, workload, right? So the chemists which are known to us, right? At least we can request them, okay, whenever some person comes to pick your expiry, right? Just hand it over to them. So through that, they are disposed of safely. This is what we can do as a layman.
Okay, sir, this is the way to manage the waste. For example, if I'm talking about, is there any suggestion that if people are talking about whatever that waste you can utilize for fruitful purpose, this kind of waste anywhere we can use for the beneficial purpose? Is it possible or not? Or this is the way to just discard it?
It has to be discarded because see,
Okay.
for example, I'm taking cetirizine, right? Now, cetirizine is antihistaminic drug, right? It let's say it's a tablet weight is if it's the strength is let's say 10 milligram, it's a tablet weight is around 70 milligram. So out of those 60 milligrams, they are the some excipients which are of no use, which are not harmful to this particular environment, or even not harmful to any body also, any living system also. But what about that 10 milligram of cetirizine? That is harmful. It is not going to do any benefit. It will only cause degradation to the normal life in in soil, in water, wherever it goes. So if these drugs are getting some effects in us, these drugs will get some more effects in the other animals also.
Thank you, sir.
Thank you, Vijay.
So I think as per the discussion, these drugs are also bringing soil pollution.
Yes, ma'am.
Like if we are not disposing them appropriately, then they are increasing the soil pollution in one way or the other.
Definitely, definitely. Anything which is not required for, see, the normal aquatic life, normal, you see, soil life, right? It is poison. See, some drugs, they are, what is a poison? Poison is nothing but a chemical substance. We know it. It all depends upon the dose. At a low dose, it can be a drug. At a high dose, it can be a at low dose it can be a drug, and high dose it can be poison. Now,
There might be some half-life of that, means for how much time it is there in the soil. But we say, I'm just that term is skipping from my mind. We always say we always use a term like half-life, or you say no, not half-life exactly, the residual period in which it is active in the soil. So is it like something is there in this case also?
there
say
are
soil or water.
There are some drugs which are quite thermolabile, some drugs which are photolabile. For example, digoxin is there. For example, penicillin is there. They are thermolabile drugs. Now, when we excrete, we dispose these drugs in open environment, they degrade. That's okay. They will degrade. But what will be the degradation product? That degradation product may be harmful or may not be harmful.
May not be, yeah.
May not be. But we cannot be sure. We can be sure only after the studies.
Yes, definitely.
Yeah. So so it is always better if we don't have any study which is supporting that it is safe. We cannot say that it is it is safe.
Yes, thank you, sir. But I don't think such kind of studies are available and have been done. No.
No
one is worried about that.
No one is worried about that. Industry or the scientists, they are worried about the stability of drugs. They find out how stable they are, right? But what will be the effect of those products, those impurities? Those are not usually studied. If they are studied from the cytotoxicity point of view in humans.
Yes. Yeah. Thank you, sir.
Yes, there is a message from Dr. Renuka Das. We at our college have a recycler club where students work dedicatedly towards recycling of paper, plastic, as well as e-waste. Yes, Dr. Renuka, that's good. At the college level, we can do it. Like Dr. Umesh, you said that as a layman, what we can do. So at institute level, we can do it.
Yes, sir. It can be our first step.
Yeah, that's good. Good idea. So if there is no query, then should we proceed? Because we have 15 minutes. Okay, can we take a break for 5 to 7 minutes?
Sure, sir.
So in the meantime, if anybody will be having any query, we will be taking that query, and after that we will start with the next session. So continuing with the morning session. So the next topic which we are going to talk about is the environmental contamination of food, or the environmental contaminants which are contaminating our food. Now, when it comes to food, the first term which we should be aware about is the food security. What is food security? So it is the permanent access to sufficient, safe, and nutritious food supply that is needed to maintain an active, healthy lifestyle. This is what we all want from the food which we eat. And this is basically our right also. And it should be the duty of all food suppliers or food manufacturers to supply safe and nutritious food, which we are buying out of our pocket by spending money. And food safety is another term which pertains to those food manufacturers or food suppliers, that they have to adhere to certain conditions. They should take certain measures which are required for the production of food, for the processing of food, for its storage and distribution, so that we get a safe, sound, and wholesome product which fits our consumption. But this food safety insurance is not a simple task, because this whole process of production of food till it is consumed, it is quite a complex network. It involves numerous interactions during its life span from its production till it reaches a consumer. And these interactions are among some physical and biological environments as the food moves from production to the consumption, or we can say from farm to table. So during this cycle, see, industry and other sources, they are supplying food, and at the same time they are contaminating our environment also, directly or indirectly, like air, water, soil, right? And these three components, air, water, and soil, they are producing food, but at the same time, they're also contaminating food through inhalation, through contact, or through ingestion. And that food, which is supposed to be nutritious, but many a times it is contaminated also, and that goes to the livestock or to the humans. And those contaminated livestock, when we consume, we again get contaminated. So this is a total contamination pathway which leads to a contaminated food. So this whole cycle, the various interactions, it can be divided into two categories. One is the contamination of the food due to the environmental influences. Environmental influences are like the you see our soil or water through which our food grows. So if that soil or water is contaminated because of the agricultural chemicals, because of the industrial waste, so those contaminants, they get incorporated into food, and that contaminated food is consumed by us. And second part is the contamination of the food during production or processing. So there are numerous steps in this food production and processing, starting from the raw materials. So raw materials can be contaminated. Then it is processed with the help of certain processes, certain aids, right? then those aids or the equipments, then the air which is in which it is processed, then the packaging material which is used for its packing, right? And then you see it's a cleaning process, right? Then supply chain, it's a storage, transport, right? So these all processes in one or the other way, they can also contaminate our food, and we again consume that food. So these processes of contamination are of two types. One is related to the environment, another is related to the production and processing. So here we will be talking about the environmental influences or the environmental contaminants, not about the contamination which is occurring during its production or processing or packaging or distribution. Now, this contamination due to environment, it occurs directly or indirectly. Directly, like if we are contaminating, if we are using contaminated water, like in the last talk, we have seen that the treated water, treated wastewater or the sludge, it is mixed with the you see manure and is used as a farm manure, and the treated water is used to irrigate crops, right? And those chemicals they get incorporated in the food. So that is direct environmental contamination. But there are certain you see environmental changes which are leading to climate change, like global warming, right? So global warming, we know this is a holistic process, right, which is leading to increase in the temperature of our environment, and that global warming causing a climate change that also affects our food quality. So this climate change is very likely to affect the global, regional, and local food security by disrupting the food availability, decreasing access to food, and making utilization more difficult. Now, the different climate drivers, the climate change drivers, like temperature and extreme heat, or change in the humidity conditions, or changes in the timings of the length of the season. Nowadays, we have seen if we look back around the 15 years or 20 years, we see a change in the length of the seasons. Winters getting shorter, summers getting longer, right? So temperature is changing, but for the crop to flourish, there is a specific time period required. There is a specific conditions which are required for its you see sowing, for its ripening, for its harvesting, right? Those climate drivers, right, they are affecting us in two ways. One is the environment, one is from the environmental context or the institutional context. If the climate drivers change, the climate change, it results in the change in the agricultural practices and the livestock processing. It results in the change in the water quality management issues, then transportation and infrastructure for the food distribution, and power outages. So these are the key factors which are you see vulnerable due to this climate change. And these key factors, they change the agricultural practices, leading to change in the food quality. Second is second type of key factors which are affected by climate drivers are at the personal level or at the individual level, like they are classified as a social or behavior in the social and behavioral context, social determinants of health. So as the climate change, our health change, the food handling practices and preparation. So as the climate change, our food handling practices also change. Increase in outdoor activities like barbecues and picnics, they are also related to the climate change. Then the lack of home refrigeration. So like to store food, we need a refrigeration, and if we don't have that refrigeration, that is again a change in the style. Then awareness of and response to outbreaks. So these are at the individual level, how we react, how we respond to these changes. These are two different key factors. One is at the individual level, and the second is at the society level or the holistic level. They change the exposure pathways. How they change the exposure pathways? Uh, they increase the growth of pathogens. Like in if the humidity is higher, temperature is higher, then the growth of the pathogens increase. Then there is a seasonal shift in the incidence. For example, in case of salmonella, like in case of if we see that bacterial infections, they are more during June, July, and August, because that particular season is more conducive to the growth of bacteria, and those microbes, they contaminate food. Then consumption of undercooked food, undercooked poultry. So that is another exposure which makes us prone to the contaminated food. And all these factors, the exposure pathways, the individual factors, or the environmental factors, they directly affect our health. If we are, because they are contaminating the food, because they are changing the composition of the food, so they are affecting our health also. See, this is a study where the seasonality of some human illness is assessed with respect to some bacteria. So over a period of year, January, February to till December. See here, June, July, August, right? September. There is a maximum infections because of the bacteria like Campylobacteriosis, then Salmonellosis, then E. coli infections, they are maximum. And the viral infections, they are minimum in this region, but during this period, but they are maximum in the winter seasons. So that's why in the winter, we all know that during winters, we get viral infections. And in the summers, or in particularly during the rainy seasons, monsoon seasons, we get the bacterial infections. So these are related to, climate. And these bacterial infections or viral infections, they are not just in the air, they are not just in soil, but they are in water. They are also in the food which we ingest. Some very common chemical contaminants in the different foods, if we see, they are belonging to the different categories like metals or metalloids, like lead, mercury, lead and cadmium, copper, zinc, uh, this titanium, then we have iron, manganese, right? They all are present in one or other type of food articles like grains and vegetables, livestock organs, then sheep livers, agricultural crops, locally produced fruits, lettuce, soybeans, fish. So different kind of food, they are contaminated with metal ions. Mycotoxins, this is a kind of chemicals which are very toxic in nature, and different kind of mycotoxins like aflatoxins, nivalenol, right? Then we have organotoxins, then so on. They are present in different kind of food articles like wheat, barley, Japanese retail food, then wheat flour, maize, cereal and cereal products, groundnut oil. So they are contaminated with mycotoxins. Then some antimicrobials or antibiotics, they are present in pork, wheat, table eggs, milk, beef, meat. See, the possibility of finding these antimicrobials is more or it is localized to the animal produce, because these animals or these livestock, they are given antibiotics or to maintain their health, And ultimately, we eat those livestock, resulting in contamination of consumption of the contaminated food. Some polycyclic aromatic hydrocarbons like benzopyran, creatine, anthracene, fluoranthene. So they are different types. They are present in barbecued foods, yogurt, grade flour, bran, ester, and these the countries which are mentioned here. These are the specific countries where these tests were carried out, but it is not just localized to Sweden. So wherever the barbecue food is there will be benzopyran. Now, from where this polyaromatic hydrocarbons coming in barbecued foods? Because polyaromatic hydrocarbons, they are usually present in wood. And when we barbecue foods in, you see, smoke or in wood fire, those aromatic hydrocarbons, they get incorporate in the food. Similarly, yogurt, then grain flours, right? So they are just not limited to particular country, which is mentioned here, but they can, if they are if anthracene is present in yogurt in Italy, it will be present in the other countries also. Then pesticides and the polychlorinated biphenyls, which are used for growth of crops, to protect them from weeds, to protect them from insects, from protect them from pests or so on, like chloropyrifos, this is a pesticide, then DDT and other OCPs, that they are again pesticides, then similarly other pyrethroids, they all are present in different kind of livestock or food articles like catfish, vegetables, food plant, milk, chicken products, baby foods, honey, cereals, or so on, right? So these type of chemicals, they are also found in different food articles. Then radioactive substances, that is also found in certain areas where this you see handling of radioactive materials are carried out in particular regions, uh, like radionucleotides, uranium, or radioactive substances in water, food, seafood, water and seafood, right? So on in Japan, India, Balkans, Switzerland. So in seafood and water, the radioactive substances are high because the reason may be that most of the radio nuclear activities, they are carried out near you see sea, and the waste which is you see released into water, right? So that contaminates water and that contaminates the seafood also. Now, these different types of contaminants like metals, mycotoxins, polyaromatic hydrocarbons, or so on, pesticides, they have different pathological effects when they are consumed. Like lead, it is very severely responsible for complication in our system. Like Dr. Kohli has mentioned in his talk, that the dye which is used to color hair, right? It affected a grandson of a lady, because that lady used to apply dye on her hair. So that lead is, you see, not only through air, it is not only inhaled, but that can be also consumed through food. Then cadmium is responsible for renal tubular dysfunction, means kidney functions, and it also makes the people at high risk of lung and breast cancer. Osteoporosis, loss of bones, that is also effect of cadmium. Arsenic is another heavy metal which is associated with the dermal problems, skin problems, respiratory problems, and they also have mutagenic and carcinogenic effects. Nickel is again associated with the skin problems, loss of body weight, and you see it also has fetal effects. Mercury is another heavy metal which is linked to cardiovascular disorders, and then reproductive disorders, then toxicity, neurotoxicity, nephrotoxicity. Nephrotoxicity is related to kidney, immunotoxicity that lowers our you see immune system, and carcinogenicity. Uh, when it comes to the heavy metals, see, these kind of heavy metals, they are specifically found high in those crops or in those regions where treated water is used for irrigation, and in those crops which are grown towards the roadside, because our vehicles, they emit um, heavy metals. Although nowadays the petrol is de-leaded, but definitely the petrol or the diesel or the fuels, these conventional fuels, they contain high amounts of other heavy metals, and during emission, those metals which are emitted in smoke, in the vehicle smoke, they get deposited on the soil, on the crops which are lying along the roads. So from there they get impregnated into the crops. Then our effluent treated effluents, they contain again heavy metals, because the sewage treatment plants, no doubt they treat the sewage, they remove certain solids, they do oxidize certain things, but many heavy metals, they remain untreated, they remain there as such. And if that water is used for irrigation purpose, so those metals, they get installed in the food on the crop. Then mycotoxins, aflatoxin, that is causes immunodeficiency, uh, liver cirrhosis, then the nephropathy, impaired intestinal integrity, hyperestrogenism, right, or esophageal cancer, reproductive dysfunction. So these are the different effects, pathological effects of the different kind of mycotoxins. And how these mycotoxins are produced, that we will see in the subsequent discussions. Then there are some antimicrobials like tetracyclines, quinolones, macrolides, sulfonamides, which are particularly found in livestock, because they need to be maintained to maintain their produce, to increase the yield, right? And these tetracyclines, they impair the intestinal flora. Quinolones like ciprofloxacin, ciprofloxacin, they are responsive for the drug-resistant pathogens. Then macrolide antibiotics like erythromycin, they cause hypersensitivity, and sulfonamides, they cause kidney damage and nephropathy. Then polycyclic aromatic hydrocarbons like benzopyrenes, these are the most common one. They cause mutagenicity and carcinogenicity. Then mutagenicity means they cause the changes at the genetic level in the fetus also. Then they cause DNA damage, they cause oxidative stress, male fertility is affected, and respiratory diseases are another effect of these polyaromatic hydrocarbons. And cognitive dysfunction means the loss of memory or the impaired memory in children. Then pesticides, which are again usually found in the crops, like chloropyrifos, neurological dysfunctions, endocrine disruption, and infertility, and fetal malformation. Then dioxins and polychlorinated biphenyls, which are again a type of pesticides, they cause language delay, they cause disturbance in the mental and motor development, and other neurological disorders. So these different types of chemical contaminants, which can come from different sources in different types of foods, they do have very severe effects on normal health. So like drinking water is the most affected food commodity, which is affected by different kind of chemical contaminants, particularly the heavy metals and the pesticide residues, like aluminium, arsenic, lead, nitrate, then pesticide residues or sulfate. And they all have different kind of pathological effects, as you can see here, ranging from see skin diseases, to mental disorders like Alzheimer, to cardiovascular diseases, immunological diseases, cancers, right? Then you see gastrointestinal problems, right? Then you see the other immunological disorders. Now, mycotoxins, or they are also called as phytotoxins. These are toxic chemicals which are produced by molds that grow on crops prior to harvest or during storage. And this happens because of change in temperature. Now, when there is a drought, there is an increase in temperature, and during this, there is you see a huge stress on the plant. That is a water stress or the thermal stress to plant, and the immunity of that plant is compromised, and that lowered immunity of the plant attracts attack from molds, and that mold produces mycotoxin. Now once the mold attacks crop, then the mold sticks there, and whenever the conditions they become again conducive, we get rain, or we get irrigation, water from irrigation, right? Temperature changes, the water comes, so warm and moist conditions occur, and those conditions are favorable for the mold growth. So once the mold stick to the food and the conditions become conducive to the growth of plant, though also become those conditions, they also become conducive to the growth of mold, and those molds they produce mycotoxins. Then post-harvest contamination is also affected by environmental parameters. Now once the crop is harvested, it has to be dried so that its moisture is brought to a level where it does not promote bacterial growth or fungal growth or mold growth. But if that drying is not proper, if that post-harvest processing is not proper, that will promote again the contamination of the food with bacteria or with the microbes, and resulting in incorporation of toxins released from those bacteria or from those fungi. Now, aflatoxin is a major mycotoxin. It is in fact a naturally occurring mycotoxin in corn. Now, corn is a major crop in Western world, and the aflatoxins, they are the known carcinogens, means they induce cancer. And aflatoxins, they can also cause impaired development in children. They cause suppression of the immunity, immune system, and if the exposure is very high, it can lead to death also. So aflatoxin contamination in corn, it increases usually with climate change. How? If the climate change is there, like with global warming, because of the global warming, temperature is increasing, there are more incidences of droughts, right? There are more irregular rains. So those conditions, they lead to inoculation of those molds in food, and that molds, they again then they produce toxins and causing the contamination of food. So these corn is specifically affected by mycotoxins. So many a times we might have seen, while we also in India, we also use this corn, right, in one or the other way. And whenever we come across this corn, sometimes we find see a small growth in that particular piece of corn, and that is what mold is. So that particular corn has to be discarded straight away, so that we don't get infected food. And the other commodities or other crops which are contaminated by mycotoxins, they are peanuts, cereals, and fruits. Dr. Kohli also mentioned in his talk about the peanuts. There are some small circular, patches around the peanuts. those peanuts, those circular patches are because of the aflatoxins. So those are the aged peanuts. Similarly, when the cereal grains are aged, they become contaminated with the mycotoxins because the molds, they grow in the conditions which are usually found in our homes. The temperature is good, like ranging from around 30 degrees Celsius, and humidity is also good. So they grow good and produce mycotoxins, which are harmful to us. Polychlorinated biphenyls, which are again a kind of pesticides which we use, which farms use in their crops to protect their crop from the pests or so on, to increase the yield. So they occur in food as a result of environmental contamination, leading to accumulation in the food chain, and that accumulation is also because of the direct contact with food or animal feed, because we directly spray those polychlorinated biphenyls over the crop. So there is a direct contact. And then there's the contact with the food packaging material. What kind of packaging material is used for to pack the food, like the aluminum foil we use, or some other foils, other polythenes we use. So those polythenes, they contaminate the food. Generally, it is said that one should not put hot food in a polythene or in any packing material. This is again the reason is, as the temperature increases, the leaching or the transfer or the diffusion of those chemicals from the packaging material to the food increases. So that results in the contamination. Now, if you see the cycle from farm to table, when the movement of farm to table, this not only these contaminants, the climate change also increases the quality of the, also alters the quality of the food. If the global warming is there, then carbon dioxide level is increasing. That's why we call it a carbon footprinting has to be done. How much carbon dioxide is released into the environment. See here, the temperature and precipitation extremes are like flooding. They can increase the pathogen load. So this is one factor. And the climate can also alter the weed, insect, and fungal population and increase the pesticide use. So when the climate change, the temperature change, right? So that with those changes, not only the agriculture, not only the food production or food yield changes, but also will also change the behavior of the microbes, and the microbes also grow with the change in temperature and change in humidity. And if the microbes are increasing in the crop, then of course we have to use more and more pesticides. So this develops a vicious cycle, right? So with the climate change, we are doing something by which the climate is changing, and those climate changes are changing the physiology or the behavioral pattern of microbes or of the livestock, of the crops. And as a result, the crops are affected, and the crops are affected means crops get contaminated. So we consume contaminated food, and we excrete those contaminated contaminants into the environment, and that further contaminates our food. So this is a continuous cycle by which we get regularly contaminated food. So this chain has to be broken somewhere. If it is not possible to break the chain, then we have to be cautious how we can minimize it. Then the pesticide residues, the increased pest pressure, as we have said, that with the change in the climate, there will be change in the temperature, change in the humidity, that will increase the pest load on the crop. So with this increased pest pressure or increased pest load, and because the pest is increasing, therefore the efficiency of the pesticide will also change. The pest may become resistant to certain pesticides. The pesticide concentration or the may not be enough to control a particular population of pesticide. So that lead to further increase in the use of pesticides, that leads to contamination in the field where we are growing the crop, and that increases the exposure within the food chain, exposure to these pesticides in the food chain. So we are a part of the food chain, right? So we are getting exposed to more and more pesticide because of these climatic changes. So the potential changes in veterinary practices, see, this is one of the one reason for contamination of the food, an increase in the use of parasiticide and other animal health treatments. Nowadays, more and more people, they are getting towards particularly the young generation, they are getting involved into the consumption of meat products, non-vegetarian products, and so the demand of non-vegetarian product is increasing, and in order to increase their yield, the producers, they use parasiticides or antibiotics for the treatment of their produce, so that their yield can be maximized, so that they can be maintained at a particular level. And so we are exposed to so the animal or the livestock, they are exposed to these climatic changes also. So this increases the risk of pesticides entering the food chain, or led to evolution of pesticide resistance. So if the pesticide resistance is increasing, so we are getting a more and more potent pesticides. So if a particular chemical is lethal to pest, it will be lethal to us also. And pesticide poisoning is very common, like a phosphate poisoning, organophosphorus poisoning. Organophosphorus compounds, they are very good pesticides, but they have very severe effects on human health also. Like Dr. Kohli mentioned in stock, the use of mosquito repellents, they are nothing but they are the insecticides or insect repellents. So they will affect our health also. So with subsequent implications of for the safety, distribution, and consumption of livestock and aquaculture products. So the all factors increase. So climate change may affect aquatic animal health through temperature-driven increases, right, in disease. Aquaculture, if we talk about aquaculture, for example, production of fishes, production of seafood, production of freshwater food like crabs, prawns, or so on. To maintain their produce, again, to maintain their yield, to increase their yield, the producer have to add antibiotics into the water. They have to add pesticides or the parasiticide in the water, so that the health of the animals which are grown for the purpose of food, serving as a food, that's not affected. So every climatic change or every change in the environment, it is ultimately leading to increase in use of pesticides, increased use of antibiotics, increased use of parasiticide, which get incorporated ultimately in the food, whether it is vegetarian or non-vegetarian food. Now, how the carbon dioxide level is affecting the crops, how they are contaminating. Here we will not say exactly the word contaminating, but they are altering the nutritional values of the crops. Every crop is known for its specific use. Some crops, they are rich in carbohydrates, some crops, they are rich in proteins, some are rich in minerals, right? So every crop has its own specific use. But if carbon dioxide load is increasing, then the protein content of the major crops is likely to decline significantly. Because if the carbon dioxide is increased, then the photosynthesis will increase. If the photosynthesis increase, it means that will produce more and more carbohydrates. So the carbohydrate load will increase and the protein load will decrease. So that will affect the ratio of the major macronutrients, means carbohydrate to protein. So carbohydrate to protein ratio will be increased, and that will directly affect our metabolism, and that will directly affect our body mass. Similarly, some micronutrients or macro elements are like iron, zinc, calcium, magnesium, copper, sulfur, phosphorus, nitrogen. In most plants, which are usually the vegetarian plants, they are also decreased because of increase in the carbon dioxide load, because the normal process is the photosynthesis, not the assimilation from roots. So they get affected, the assimilation get affected. As a result, these nutrients which are obtained from soil, they get affected when their nutrition value is compromised. So this is the study where an effect of carbon dioxide on protein content in wheat flour is studied. As we see here, when the atmospheric concentration of carbon dioxide is 300 ppm, the protein content is, see here, around 19.2, 19.1. And as the carbon dioxide load is increasing, see here, which is a projected one, right? By 20, because here in 2000, this study is carried on 2014, right? So in 2008, the carbon dioxide load was around 385, and the protein content was less, around 18.6. And here in 2050, 2010, it is projected that if the carbon dioxide load goes beyond 715, the protein content will be very less. And similarly, there is a concentration of the essential minerals at carbon dioxide concentration at 689 ppm. See here, this is relative to the control value, right? Control value. See here, minerals, they all have decreased. There is a decrease in the mineral content with the increase in carbon dioxide, like nitrogen, phosphorus, potassium, calcium, sulfur, magnesium, iron, zinc, copper, manganese, right? So they all decrease. Protein content also decreases, like barley, rice, wheat, potato, soybean, which is considered the main source of protein. Even in that, the protein content is reduced. So even in soybean, which is mainly known for protein, in that, with the increase in the carbon dioxide, the protein concentration is decreasing. So climate change is affecting the nutritional value of the food also. So here I will present you a case study which we have carried out in our laboratory on milk and meat. We say that veterinary produce, the livestock, they are given drugs, antibiotics mainly, and the painkillers. But they are also given some steroids, the reproductive hormones, to promote their growth. For example, chicken. If we talk about chicken, the quality of the chicken depends upon how fleshy it is, right? And also in the poultry farms, the poultry producers, they tend to increase the yield of the poultry, means more chickens, more income, right? So they give several steroidal drugs, reproductive drugs, like dithydrotestosterone, progesterone, hydroxyprogesterone, testosterone, norethisterone, estradiol, right? Some of them are natural and some of them are synthetic one, so that their produce can be maintained at a to give the specified yield. Again, this is about the chicken. But when we talk about the veterinary animals, like buffaloes or cows or goats, they are also given these drugs to increase the milk production or to increase the body mass, particularly in goats. These chemicals, these are drugs. As we said that our body can eliminate all the drugs unchanged up to 90%. So 90% of the drugs, they are excreted. So they are excreted not only in urine, but they are also excreted in milk. So that's why you see that when a mother is lactating, so it is recommended that she should not take any potent medication, because that medication may get excreted in the milk, breast milk, and that will directly affect the newborn baby. So there are these drugs which are excreted in milk, and that milk we are actually consuming. So we carried out this study, and we to see that what is the different what are the concentrations of different type of these drugs or hormonal drugs in the milk, and to what extent and how it varies with the menstrual cycle of the life cycle, or the pregnancy cycle of a particular animal. So with the help of chromatography techniques, we tried to identify and quantify different kind of these drugs. So here we see that this is a standard drug, and these are the drugs which are noted in milk. So here see the testosterone and hydroxyprogesterone and testosterone, here another. So there are two derivatives of testosterone propionate and testosterone isocaproate. So these three drugs are found in milk sample, right? So these are the standard drugs, and these are found in the milk samples. This is another sample in which we see that here it is estradiol in addition to the other drugs which are analyzed, and see here this one red color is a standard one, and blue color is a milk sample, and here 12 number testosterone is found in the milk sample. So this is the concentrations of different drugs which are found in milk samples. So here E2 is a ethinyl estradiol, norethistyrone, diethylstilbestrol. Diethylstilbestrol is a synthetic steroid which is not produced in the body. It is given from the outside only. Then progesterone, which is a natural hormone. Then medroxyprogesterone, it is again a steroid, a synthetic steroid, which is given to humans to prevent pregnancy. Then testosterone also there, and see the concentrations. So the concentrations on the different animals, we can see it's around 13 microgram per liter of ethinyl estradiol. Then progesterone is present. In some animals, diethylstilbestrol is present. The norethistyrone is also present, right? So these drugs are found at microgram per liter or sub-microgram per liter levels. So similarly, the other samples. So I have put all the samples here just to give you a view that how the content of these drugs, they vary. Like ethinyl estradiol, it is a more or less same in all the drugs, in all the samples, around 2.8. Right, norethistyrone, in some it is absent, in some way it is present. Diethylstilbestrol, in some one it is present, some way it is absent. So if it is a diethylstilbestrol is detected, medroxyprogesterone is detected, it means something is given from the extraneous source to these animals, which is being excreted in milk. And this milk is consumed by all age groups. So these are other samples you can see here. And so this is other type of milk, right? See here in all the cases, they see ethinyl estradiol, norethisterone, diethylstilbestrol, progesterone, medroxyprogesterone, testosterone, they all are present in milk samples, different milk samples. And how this milk concentration, these drug concentrations vary during the pregnancy. See here, this is the postprandial, the postpartum period, where the animal is not pregnant, right? So as the pregnancy increases from non-pregnancy to trimester 1, trimester 2, trimester 3, and we see that there is a rise in certain steroids, like estradiol is increasing, progesterone is increasing, and the testosterone is decreasing beyond T1. These are the natural, you see, hormones, and these hormones, their behavior is similar to those observed in humans, right? So in human also, this kind of behavior is seen. So they are the natural one. If we say they are the natural one, so they should not affect our health. But when it comes to the synthetic one, like norethisterone is there, medroxyprogesterone is there, diethylstilbestrol there, right? They are the non-natural one, and see, their levels are controlled. Their levels are varying. Why their levels are varying? Because their dose is controlled by the producer, so as to maintain the pregnancy. So in order to maintain the pregnancy of animal, so that the milk production is increased, right, they give the extra drugs, medroxyprogesterone and diethylstilbestrol, so that the milk production is not compromised. So similarly, analysis in the meat samples, like in mutton, which we call it goat meat, right? See here the concentration of ethinyl estradiol, diethylstilbestrol, progesterone, medroxyprogesterone, testosterone. See here the concentrations, 39 micrograms per kg, right? Testosterone is there, right? Then chicken breast, which is a very, these are the two parts of the chicken breast and chicken thighs, which are most commonly eaten, consumed by the, you see, person, right? So chicken wings, chicken breast, they are, see here, rich in the testosterone, but they are very less or almost absent in the other hormones are absent. But chicken thighs, they are high in, see, ethinyl estradiol, then diethylstilbestrol, progesterone, and testosterone. So these are the parts which we consume. Then chicken wings, which are specifically, you see, cooked or supplied by certain suppliers, right? And they are, you see, sold at a very hefty price also, and they are also rich in see, different hormones, like testosterone is there, medroxyprogesterone, progesterone, or so on. So when we are consuming these meat samples or milk samples, we are consuming these hormones also, and these hormones, they affect the normal reproductive system. So some studies are carried out on these animals, like in the last talk, we had learned about the fish, how ethinyl estradiol, it has affected the fish growth. Then if it has affected the fish growth, it will affect the humans also. And it is also studied that what happens during boiling. If we boil the milk, right? So the milk boils at a temperature or at around 120 degrees Celsius. Now at 120 degrees Celsius, these components should degrade completely because it's a high temperature. But we found that these drugs, they did not degrade completely. They did degrade, but to a lesser extent, not more than 20 to 30% degradation was noted after the boiling of these this milk, or right. So if the meat is cooked, we don't take raw milk, and if we take you see cooked meat, right, even in that meat, the significant amount of these hormones will be there. So these hormones are affecting our normal reproductive cycle. If we compare the growth of normal teens nowadays vis-a-vis the growth of teens around 30 years back, right? We see a huge difference. The growth of secondary sex organs, the growth of, you see, the reproductive organs, you see the behavior of the human beings, right? There is a dynamic shift. There is a shift. So whatever the changes they were usually seen and at around 16 years of age, around 30 years back, those kind of changes are nowadays seen at the age of 13 years or so, right? So what are the reason of those changes, right? I don't say these are the purely contributed by these milk or meat, right? But definitely we are getting more and more steroids, these drugs from one or the other source, which is affecting this growth pattern. So what is the, you see, solution to this? How we can prevent or minimize the food contamination, right? So many things are not in our hand basically, but there are these remediations, they are more and more, you see, concentrated, or they are more concerned to the producer or at the government level. Like first is the soil remediation. If we change the soil, like remove the contaminated topsoil from agriculture areas, so that is the best thing we can do. But this is a very expensive affair and no one wants to do it, because to remove the topsoil layer, covering it with a new one, right? So that cost, that increases a huge cost and no one will do it, right? So the alternate is we can turn over the soil, right? So that the contaminants which are on the topsoil, they get diluted by mixing with the soil underneath, right? And in the meantime, some of the contaminants, they will get to the bottom, right, of the earth or bottom of the soil, right? So that they get not impregnated in the food. Then thermal treatment or landfill can also used to remediate a site. Thermal treatment, that is again an expensive affair. Then microbial bioremediation, that is another way by which means we can mix, we can inoculate certain bacteria in the soil, and those bacteria should be able to degrade these contaminants. So that is a good approach which we can use. Then phytoremediation. There are certain trees which attract certain kind of contaminants. For example, black nightshade, also called as Solanum nigrum, which is usually you see planted around the tree on the crops, and they remove thallium from the soil. Now, thallium, which is heavy metal, right? It is absorbed by Solanum nigrum, right? So as a result, the soil becomes less contaminated with thallium. Then crop management techniques can be there used at the you see agriculture level. So agriculture practices have to be changed. For example, use of a slow release nitrogen fertilizers can reduce cadmium level in some plants. Then we can change the water management. We can adopt the water management techniques like drying the paddy field for a period of days during specific stages of its growth. It reduces the cadmium and arsenic levels in rice crops of different rice species. So because you see, during growth of the plant, absorption of certain elements increase, and during that period, if there is no water is a dry, then that absorption will not take place. Then safe storage alternatives that we can do at our level using edible films or edible coatings. Rather, if we can avoid the storage alternatives, means we can avoid the storage film, that is the best one. But still, if we have to use, we have to use the one which are edible and which are safe to our system. Then safer cooking practices. And although most of the people now they are following safer cooking practices, means they have shifted from the cooking on an open cooking to the cooking using gas stoves, but still, if we are going for the barbecue, for the you see, for some parties or for some outings or some picnics, right? We should avoid those barbecues, because that is the maximum main source of incorporation of a polyaromatic hydrocarbons into food. So these are the various techniques by which we this food contamination can be minimized. But the best one is the attitude and awareness among ourselves. If we are concerned about our health, then definitely we can minimize this contamination. If we are aware about it, we can minimize this contamination. Then there should be more stringent food regulations. Although in India, we have FSSAI, right, which regulates the food quality, but those regulations are not very stringent, right? So they need to be more stringent. With all, thank you so much, and have a happy and healthy eating. So if there's any query about this, what the food contaminants? Any query, please? So if there is a no query, then should we split for lunch?
So if any participant wants to ask any question, he can ask Professor Gulshan in the chat box, or he can write email, or discuss it on the telephone, as you like. And we are available for all the times to you. And it was a very nice presentation, Professor Gulshan, and earlier one also. And it
Thank you.
seems too difficult to eat anything, and that we have to be very careful about eating.
Yes, we have to be careful. We have to be careful at our level only. We have to protect ourselves.
Yes, only then we can survive.
Yeah. Okay, then we split for lunch, sir. We will meet
Yes, sir.
again at 2 o'clock. So all the participants are requested to join by 1:55 p.m. and the next session will be by Professor Yolanda. So she will be talking about contaminants of emerging concern, and then she will be talking about handling of this how to characterize this environment contaminants of emerging concern. So see you at 1:55.
Yes, sir.
Of this. Regarding the categories of brominate flame retardants, you have two types of these brominates. Ones are the additives that are mixed with polymers but not linked chemically to the plastic, like for example, the polybrominated biphenyls and the polybrominated diphenyl ethers, and instead there are others that react with the plastic, that are the tetrabromobisphenyl A, the TBBPA. Then this is important because these compounds, has been used in the forest fires, of course, years before, but now they are restricted to textiles and to closed places. Many textiles from the buses, from the planes, etc., are impregnated of these compounds with the idea that in the case of an accident, they don't get burned, and then to preserve better the human. Here you have in this slide the different structure of these compounds. You could see the similarities. the polybrominated biphenyls are equally to the polychlorinated biphenyls. In fact, they can have 209 congeners because the biphenyl ring can have a variable number of atoms of hydrogen substituted by bromide, and if you make the combinations of how many possible structures do you get, you will get 209 in the case of the polychlorinated, and in the case of the polychlorinated of the polybrominated biphenyls, and then you have also the other structures just to make you familiar with this type of contaminants. Here you have a table that shows how many tons of this compound has been sold in the European Union before their effective ban. These compounds in the European Union were banned in 2003. In the rest of the world, depending on, in many places they hasn't been banned, but in any way there is a restriction in the use or fabrication. In many cases, this has been done as an agreement with the industry, because nowadays economy is very important, and to keep the working places of the people also. And then there has been an effort to, let me say, make things friendly. But there is a kind of being a restriction in the fabrication everywhere. If you could see, in the moment that the penta biphenyl brominated diphenyl ether was banned in Europe, you see that the use has been fall down, but the use of many others has increased. Then that is a problem. Regarding the environmental levels that we can find of these compounds, PBDE concentrations is between has been reported between 350 and 2,300 micrograms per kilogram in a purpose bubble in Canada, and between 22 and 340 in salt. Then depends of the animal species, on the fat content. In that, for example, porpoise bubble is a oily fish, and then it has a higher fat content, and then depends on the fat content, because these compounds tend to accumulate there. And well, you have here a few data about how many micrograms per kilograms of these compounds has been found in several fish species everywhere in the world. Then, as you could see, the problem of these compounds is very similar to that of the classical persistent organic pollutants. In fact, this group, together with the next one, the perfluorinated compounds, are considered as the emerging persistent organic pollutants. Regarding their effects, they have a neurobehavioral effect, effects on the thyroid hormone homeostasis, effects on the liver, and penta-BDE seems to cause adverse effects at comparable lowest doses than other compounds. It's considered that about 0.15 milligrams per kilogram of body weight a day doesn't have any effect, and there is a proposal of tolerable daily intake of 0.15 micrograms per kilogram of body weight in the EFSA. You have here, for example, one estimation below the journal where I took this estimation, and also the method that was used to make it. And you could observe that one of the problems of these compounds is that if you look at several groups of populations, the exposure of adults to these compounds through skin, diet, and breath is really low. But instead, if we go to the upper file, you will see that for babies it's very high because they tend to be accumulate in the human milk, in that the human milk has a 3% of fat, and then this is a kind of lipophilic compound that tend to accumulate this organic content. Then that is the problem with these compounds. You have an idea that we have substitute one for others, but really don't solve the problem of contamination. The other compounds that are nowadays very dangerous and that cause concern everywhere are the perfluoroalkyl substances. The perfluoroalkyl substances former were called perfluorinated compounds, but nowadays more or less the nomenclature rules are going through the perfluoroalkyl substances, and these perfluoroalkyl compounds are constituted by a large group of compounds that are characterized because they have a fully fluorinated hydrophobic linear carbon chains that have attached one or more hydrophilic head group. If you see, you have in this slide one of the most popular perfluorinators that are the perfluorooctane the perfluorooctanoic acid. You see that it's a very long linear carbon change of where all the hydrogen atoms has been substituted by fluorine. I have to tell you that fluorine is one of the most strong or the most stable bond, because the energy that needs the carbon fluorine bond to be formed is 485 kilojoules per mole. Then if you look at this, these are very stable compounds. And in addition, if you look, maybe I have put this energy, sorry, I forgot to explain it. And below you have the energy that is needed to form other bonds in order to give you a comparison and to give you an idea of how much stable is this bond. These perfluorinated compounds, if you look at it, just what I have put in this slide, you will see that they remember slightly surfactants because they have a very long linear chain that is non-polar and a polar head. Then this is true, they are in part behave like surfactants, and this is also a property responsible of their interesting properties, and also that makes them to be compounds that has been highly used in many places. You have other types of perfluoroalkyl substances. I have shown you the perfluorooctanoic acid, that is the PFOA, and 4 parts of the first group, perfluorocarboxylic acid. Then you have the second group, that is the perfluorosulfonic acids. The equivalent to the perfluorooctanoic acid would be the perfluorooctane sulfonate, PFOS. Then you have also fluorotelomere alcohols that have this structure, telomeric acid, sulfonamides, phosphates, acids, phosphinic, etc. Then there are many structures. Commonly, these compounds degrade from polymers like, for example, Teflon, and the endpoints of the degradation are the PFOS and PFOA. And to give you an idea of what are the most abundant in the environment. These compounds are has been used in many places in the covering of the frying pans, were very normal. in some clothes that were made of Gore-Tex. Gore-Tex is still in the market, but it has changed the composition, and nowadays they don't have PFOS or PFOA, but a few years before they use it because this compound has interesting properties repelling both water and oil. Then they have also in use in the packets of food. For example, this food that is ready to prepare in the microwave oven has had years before many PFOAs, and also in electronic components, stain repellents, textiles, paints, waxes, polishes, electronics, adhesives, food packaging, etc. Nowadays, probably they has been changed, but years before the textiles that we use to eat that were very modern, they are repellents of any type of staining, were made from this compound. And this is because of their interesting chemical properties. They are thermostable, could support temperatures higher than 150. They are resistant to degradation by acids, bases, oxidants, etc. They are hydrophobic and lipophilic at the same time. In fact, if you look for this P or constant of distribution that I showed you yesterday, the main problem is that cannot be calculated experimentally because they form three phases in the mixture octanol water, because this characteristic are very good surfactants and lubricants, are non-flammable, chemical inert, and as I told you before, repel both water and oil, and therefore they are ideal for surface treatment. Then I would like to tell you that we are not bad with nature. That means that we don't contaminate because we want to contaminate. We do it because we fabricate chemicals that give us a very comfortable life and has very nice properties to make life easy. These are what I would like to say you about perfluorinated compounds. But the problem is that they are toxic. Then studies indicate that PFOS and PFOA can cause reproductive and developmental, liver and kidney and immunological effects in laboratory animals. Both chemicals have demonstrated to be able to cause tumors in the animals of the study. And their results show consistent findings with the human epidemiology studies. Then they have other effects, like, for example, increase the cholesterol levels among exposed population. And well, that is more or less what is demonstrated. And then we have, but there are scientifically a lot of findings that are not fully demonstrated and that I relate to infants that birth with low weight, effects on the immune system, production of cancer for PFOA, and disruption of the endocrine disrupting effects, like, for example, over the thyroid hormone. This is for the case of the PFOS. Then not everybody knows about the perfluorinated compounds. PFOS, as you saw in the class of yesterday, has been banned and has been included in the list of the Stockholm Convention. And PFOA is not, but PFOA, for example, is not yet included, even that this compound has also demonstrated very bad effects. This compound has not been really banned, but there is an agreement with the industry to reduce their use drastically for 2020. But the main problem that has happened with these compounds is that they have so nice properties that mostly in the industry, what has been done has been to replace one perfluorinated compounds by others. Then now the industry is using the short chain perfluoroalkyl substances, like for example, instead to use the perfluorooctanoic acid, to use the perfluoroxanoic acid, and then they are, let me say, blog telling these compounds aren't toxic, but also I can tell you that in the European Union there is a call to perform research in order to substitute any the materials based on perfluoroalkyl substances by materials based on any other type of chemistry that don't involve these toxic compounds. I have shown you a few data about what happened with these compounds. You could observe here, for example, this is what happens in the drinking water of several places in the world. I include those that I find data. And you could observe, like, for example, in some places like Germany, like some places also of Spain, the amount in drinking water is very high. This has been related with studies that has been done in places where they had near some industry of these compounds. These compounds, one of the problems that they have is that are a kind of emerging persistent organic contaminant, but really they don't feel the behavior of these compounds. They, for example, don't solubilize in the lipids, but in the proteins, and they are highly soluble in water. Then one of the problems of these compounds is that once they have reached water, it's very difficult to eliminate them. In fact, there are some studies of how to eliminate perfluorinated compounds from drinking water, but these studies are based on the use of very expensive solvents like carbons and nanofiltration and these types of process that gives very good results, but are highly expensive. And in the cases in which the contamination is at very high levels, really the cleanup and the removal of these compounds is still a problem. Here you have also some concentrations in other environmental and food products, just to give you an idea about our routes of exposure. There is no full evidence of this, but in many places it's established that these compounds are passing to the human being mostly through fish, because these compounds has been since 50 years ago, this compounds has been happily released to the water and sea, and then fish is considered as one of the sources of these compounds. As you could see, if there are several food samples from the market, not only fish, and as you could see, the contamination of food products is also global all over the world. The European Food Safety Agency has established tolerable daily intake and has also write an opinion about the perfluorooctane sulfonate and the perfluorooctanoic acid and their salts, about a scientific opinion of the panels on contaminants in the food chains, where they decide that there are very little data about these compounds and has established a tolerable daily intake that is of 150 nanograms per kilogram and per day for PFOS, and and of 1,500 nanograms per kilogram and day for PFOA. I have to tell that last year there has been a very big movement against perfluorinated compounds in Europe, and that about December, January, almost all the legislation is changed, and nowadays they are not included in the priority list of contaminants yet, but the drinking water legislation and the EFSA are modifying these values. And for example, in the case of the drinking water, and I consider this highly important, there are lists about the tolerable levels for many more perfluoroalkyl substances, not only for PFOS and PFOA, but almost for all the list of potential acids and sulfonates that can be released to the water. And this, I think that is a good idea, that will increase quality, because there are many of these compounds in the environment. I don't know, well, here you have a calculation of the dietary intakes for PFOS and PFOA in several countries, just to give you an idea about the intake of the people. Groups of populations that eat highly amount of fish are considered as risk populations. And with this, I will finish with the perfluorinated compounds. These two emerging contaminants would be what is called emerging persistent organic contaminants. I don't know if you have any question. If you don't have any question, I'm going to pass to the next group of compounds that are the endocrine disrupting compounds. The endocrine disrupting compounds, well, are defined as compounds that in one or other, sorry, Sharma?
Yes, ma'am, good afternoon. Ma'am, I have a question.
Please do it.
Ma'am, I wanted to know, since you are talking about these polymerization products, so how can we prevent ourselves from the consumption of such products? I mean, is it because this is all related to our day-to-day eating habits and everything? So what measures can we take?
Well, I think that the measures are slightly complicated, but commonly, if we want to avoid this compound, it's very difficult. It's very difficult because we are in this life and they form part of everywhere. Food packaging, they are in the fish, and you don't know. Many times nowadays the market is very global, and then you go to the market and buy fish, and you don't know where it is fishing. Could be in Canada, could be in India, could be in Spain, and you don't have any clear idea. I think that the policy now is good. That means that they are trying to eliminate the production of these compounds. Then I'm sure that if the production is stopped, they will disappear of the environment in 10, 25 years completely. And of course, in a few years, the concentrations wouldn't be of concern.
Definitely. I hope this will happen soon.
I'm also looking at how is the world. I have to confess that I'm always optimistic.
Right.
But
Thank you so much.
thank you. Well, any other question? Well, if not, I'm going to pass to this new group of endocrine disrupting compounds. The endocrine disrupting compounds are compounds that in any or other way alter the hormonal system of the animals and humans. And we have two definitions because scientists it's very difficult to put in agreement with each other. And then the European Commission has a definition of endocrine disrupting compound. That means that an endocrine disruptor is an exogenous substance or mixture that alters functions of the endocrine system and consequently causes adverse health effects in an intact organism or its progeny or subpopulations. That is what the European Commission said. Instead, the United States Environmental Protection Agency talks about an exogenous agent that interferes with the synthesis, secretion, transport, binding action, or elimination of natural hormones in the body that are responsible for the maintenance of the homeostasis, reproduction, development, and or behavior. From my point of view, both definitions are, let me say, are saying the same with different words, but you see that one could be slightly more restricted than other, etc. In any case, they are composed that alter the human system. Commonly, well, substances that can interact with the endocrine system and that disrupt the normal function can act in several ways. Because they act as the natural hormone binding to the receptor, and this causes a similar response in the cell that is known as agonist response, because it's shown like if there were higher levels of hormone, or when they can bind to a receptor and avoid a normal response. This is known as agonist response because it's like if the levels of hormone were low. Also can alter the synthesis and metabolism of natural hormones, and also can alter the synthesis and functioning of the hormonal receptors. And this is just to give you a short. Important about the endocrine disrupting compounds, the effect. They decrease fertility and growth, alter sex, produce poor hatching on or thinning of the cells of the eggs, and abnormal thyroid function. Then some examples include lower reproductive, include reproductive effects on the bacterial cells, thinning of the egg cells of birth, decrease in the reptile populations like frog and crocodiles. I have to tell you that one of the most important effects of the endocrine disrupting compounds is in the reptile population, because they are highly affected. Also have effects on fish reproduction and development, on the development of male sexual organs and female marine organisms, such as snails, and also aquatic animals are the most affected are the carnivores, mainly because they are the top of the food chains. Then I'm talking already about compounds that can be bio-magnified. What are the potential effects of these compounds in the human being? Well, depending, they can present newborn malformations, undescended testicles, abnormal sperm, low sperm count, abnormal thyroid function, possible breast, testicular, and prostate cancer, and many other effects. I have put in the title of this slide clearly potential. Why? Because it's very complicated to demonstrate that these effects has been produced by the endocrine disrupting compound. Why? Because these are effects that have concern, let me say, to the people who manage health, because are effects that appear increasingly in the human population everywhere in the world. Has been known that in the last years there is a trend to increase all the effects listed here. These effects can be perfectly produced by endocrine disrupting compounds, but epidemiological studies cannot demonstrate that a clear link between these effects and the presence of endocrine disrupting compounds. Cannot show a clear effect, but also cannot show that there isn't, that the effect is not produced by these compounds. But you know, it's very complicated. It's not ethical to make studies with human people, and then is highly difficult to demonstrate. In any case, clearly, I hope that all this talk, when I explaining you what happens with the compounds, what are the most important problems they produce, what are all these things, at least demonstrate you that we are living in a world which environment has a mix of toxic compounds that we really know very little about their effects. What happens if we, instead of being intoxicated by polychlorinated biphenyls only, are intoxicated by a mixture of these compounds? Then we have to be cautious, and I think that we cannot say that contamination isn't affecting human people in any way. Well, I'm going to talk to you about the most important types of endocrine disrupting compounds. This part of the class is very easy, and I will be very rapid because many of them have already been mentioned here. For example, brominated flame retardants are important endocrine disrupting compounds, and you already know what they are. Polychlorinated compounds like PCBs, dioxins, and furans are also clear endocrine disrupting compounds, and also as compounds that we are most in contact with them because they are everywhere. We have also phthalates and bisphenol A. Don't close the idea of the endocrine disrupting compounds because, as you see before, looks like has also endocrine disrupting compounds, not fully demonstrated yet, but probably they have. And when we finish with endocrine disrupting compounds, we talk a little about the different types of pesticides, and many of them are also endocrine disrupting compounds. Then keep a little in mind this important group of compounds because they are well, important, and also can have important effects in the human being. Why? Because we are exposed to them along our life. This makes it difficult to clearly establish the relation toxicity or agent effect, but clearly is unhealthy in the world. I'm going to show you a little the other two that we don't talk about this. Phthalates. Phthalates are a kind of plasticizers that are used in the manufacture of many plastics like polyvinyl chloride, epoxy resins, cellulose esters, adhesive formulations, medical cosmetics, food packaging, etc. Then you have here the structure of the phthalates. You could see that they have this structure, and depending on the substituents earned and of the substituents of the oxygen, you can have different structures. For example, if both substituents are methyl groups, you will have the dimethyl phthalate. If both substituents are ethyl groups, you will have the diethyl phthalate, etc., etc. Then one of the most abundant is the bis-2 ethyl ethyl phthalate. This is everywhere because these are substances that are used to produce plastics, and then they have beneficial effects. In fact, the bis-2 ethyl ethyl phthalate is like a kind of punishment if you have to determine, because it's everywhere, just in the small ring of Teflon that protects the organic solvents in a laboratory. There is a big amount of this bis-2 ethyl ethyl phthalate. It's very difficult to find a blank everywhere these compounds is at high concentration. Sometimes it's in your blanks at higher concentrations than in your samples. It's really complicated to determine these phthalates because they are so widely used everywhere that makes it very complicated to determine. Here you have a few data about the use of This phthalate, you could see that this diethylexyl phthalate is about the 51% of all the phthalates found in the world. Its production is estimated in about 4 millions of tons per year. And keep in mind that 90% of these phthalates are used as plasticizers. That means that they finish in consumer goods that all of us has a big act. Of the dozens of phthalates synthesized, the most prolific in Europe are, of course, this diethylexyl phthalate. I suppose that everywhere would be like this, and the others that I have shown you in the table. An important source of human exposure to phthalates is food, especially these fatty processed foods that has been in contact with many plastics before to eat, that sometimes they are already processed and you can put in the same plastic within the microwave, etc. And the human exposure to phthalates is very difficult to assess. Why? Because as they are everywhere, their analysis is extremely complicated. Not difficult from a chemical point of view. Clearly, they are quite compounds that could be easily determined by quick chromatography or gas chromatography. The problem becomes when you want to get a blank in order to establish the quality of your measure. Then, in any case, the human exposure to phthalates can be evaluated looking at the human metabolites. These human metabolites are specific. These only appear in the human urine, and then they are a clear example that they become from original phthalates that we have that we have been in contact with. That is very important because these are, let me say, like ways to solve problems of contamination, because we would like to go to determine the original compounds, really would be highly difficult for us. You have here an example of these metabolites. Phthalates monoesters, the oxidized metabolites, and they are determined in urine without any problem of plant contamination. Of course, you always can have a contamination in the laboratory, but commonly, if you work properly, you don't have any problem with these compounds. And well, you have here, I don't go to the detail because maybe some of you are interested, some others no. This is how the phthalates are metabolized in the human body. Could be of interest, but I don't want to detail. It's all the roads that they can follow in the human body and all the metabolites that they can form. All of them can be found in urine, but of course, one are more abundant than others. And here you have different examples about the amount of these metabolites that are found in urine. Then you have here the levels in several urine samples. These are the first column. The first column, this one of here represents the metabolites that were formed in urine samples in 2002. This is for the values for the 50 percentile of the population. That means that more or less half of the population show these values. And this is the values for the 95 percentile. That means that this will cover the highest values of the 95% of the population. And as you can see, these values are in micrograms per liter, and then Some of them, these metabolites are very frequent, but many of them not so much. Then this is an important aspect of this metabolite. You could observe that this data comes from different years, and then these show differences also in the use of phthalates, because some of them are being restricted. Again, but is more or less the same play. The industry shows that the way to reduce the use of one phthalate is just to use other phthalate, and then this don't offer a solution regarding the environmental problem. The other compound that I would like to show you is the bisphenol A. The bisphenol A is a plasticizer. You see that sometimes it has been very important and very recognized because it's has been found in the baby bottles, and commonly we are more concerned about things that can affect our children. But bisphenol A is also a plasticizer that can be present everywhere. The high production volume of this compound, this compound is widely used in the production of polycarbonate and epoxy resins, and also is an additive of the polyvinyl chloride. The human exposure to bisphenol A includes indoor air, dust ingestion, contamination of food, and bisphenol A has an anti-androgenic effect because this bisphenol binds to the estrogen receptors. Both has been shown that in vivo and in vitro exposures to bisphenol A in rodents significantly decrease the testosterone production. It's used in a range of consumer products such as water bottles, sport equipment, CDs, DVDs, etc. Has been during many years an important concern. BPA contains epoxy resins that are used to line water pipes, squatting, food, beverage cans, in thermal papers. And the bisphenol A is also very well in our world, and even that it is its use has been reduced in plastics that are used to pack food. There are many other places where we can be exposed. BPA has been also used in dental sealants, and it has a commercial use since 1957. And is listed in the OECD list of hazardous chemicals. The worldwide production of these compounds is more or less established in 1 million tons per year, and more or less 100 tons are released into the atmosphere each year. Then this gives you an idea that we cannot be outside these compounds. It should be that they this production would be reduced. Here you have different tolerable daily intakes or acute reference doses that has been established by different environmental agencies. These commonly serves to establish migration limits. That means that, for example, you can observe how much bisphenol A is released for food, and then this is something that is controlled. And as you could see, if you look at the legislation everywhere, this maximum migration limits has been reduced because of the problems with this substance and the high exposure levels of the population. According to the last report of the EFSA, the current daily dose of exposure to BPA has make to reduce even more the tolerable daily index to 4 micrograms per kilogram of body weight and day, and the migration limits has been reduced as 0.05 milligrams per kilogram of food, and this looks like doesn't have a very big risk for the population. And instead, recent meta-analysis has found a relationship between early exposure to this BPE and hyperactivity in children. And then this is one of the things that has produced a high concern in the populations, because we have not only to preserve ourselves of the exposure, but also to preserve towards infant, children's lactants from the early exposure to this compound, because this can be an important cause of problem in the future. Then these are the last comment about the endocrine disrupting compounds. I don't know if you have any question. I beg you your permits to drink a little of water, because I have almost an hour talking, and then and I have passed this morning some stress with the problems with internet connection. And then I'm going to follow but other compounds that are considered also methane contaminants, like, for example, pesticides. Pesticides means any substance or mix of substances or of chemical or biological ingredients that are intended for repelling, destroying, or controlling any pest or regulating plants. Pesticides are very dangerous, and I told you this yesterday, because we spray them intentionally in the world. Then they are not only pesticides. Commonly, they also include plant growth regulators, defoliants, desiccants, agents to prevent premature fruit from falling, substances apply against deterioration during the storage and transport, etc., etc. But the most important aspect of pesticides is this wide spraying over the world. Making very, very small history on the use of pesticides, that sometimes is very interesting to understand how they has been developed, and how the criteria to develop them has been changed along the time. The old pesticides has always been used by the human being, because well, one of the problems is since the Neolithic, in which the human being start to cultivate food, pesticides has always been used, because one of the concerns of the human population has been the destroy of the harvest and the lack of food. Then from ancient times, hemlock, nicotine, and other natural substances have been used to eliminate pesticides. These are considered as natural botanicals. They are not too effective, because you need to use a very high doses, and they are natural organic compounds that are very rapidly destroying the environment, and then this gives several. Also, of course, inorganic pesticides has been used since ancient times, as the other ones, like sulfur and arsenic. Arsenic is no more used as a pesticide in the field crops, but for example, sulfur is still highly used, and also copper, especially in the wine years. And also they comes from very old times. However, the starting point of the pesticides was the rise of the synthetic organic chemistry. The first pesticide that was synthesized and used, I already mentioned, was the DDT. The DDT, was already synthesized at the beginning of the 20th century, but it started to be used especially in the Second World War. And since then, the industry of organochlorine pesticides was very, very important. However, you know, in a moment, rise the concern that these compounds were very accumulative and that they should be bad. Then the synthesis of pesticides go ahead, and the second group of pesticides that were synthesized were the organophosphates. I will show you the structure of the organophosphates, but they are esters of phosphates in which the hydrogens has been substituted by the organic molecules. We have insecticides such as parathion, malathion. They start to be synthesized in the Second World War, but and also they were more or less proof in the Vietnam War. You could see these insecticides are highly related to the European wars. These compounds, the difference between organochlorides and organophosphates is that the organochlorides were relatively little toxic and highly persistent, and instead these compounds has a strong acute toxicity, but they are less stable, less chemically stable, and then disappear in the environment very rapidly. Also, other research line that has been followed with this insecticide has been to create molecules from these natural hemlock compounds. These natural hemlock compounds were the pyretrins, and then there are synthetic derivatives of the pyretrins that try to make this molecule more stable, adding different substituents, and that give that are called synthetic pyrethroids. These synthetic pyrethroids are still highly used. The problem is that if you make them more stable, you are increasing their toxicity. And then finally, the idea has become to create molecules toxic, but really little persistent in the environment, and this gives place to the carbamates. The carbamates are characterized, as the name indicates, by the presence of a carbamic bond, but the most important problem of these compounds is that they are quite unstable in the environment, and then there are things that should be kept in mind. Pesticides are designed to keep pests. Pesticides are expensive, and in the agriculture, you have to add very high amounts of a compound that is very expensive. Normally, the agricultors prefer to use other types of compounds. That is the main problem of the carb. I have also added in this slide several milestones that has been some importance within the synthesis of pesticides. There are well known, and I don't go to detail them because, well, I would like to keep, let me say, these classes in time, but not too much time, because we have still a tutorial, and then I don't want to tire you too much with this history about pesticides. Pesticides are widely used everywhere in many things, not only in agricultural applications, but I would like to note that 80% of the the use of pesticides are agricultural applications. Then there are other use, like for example, domestic use. Also, they are used in livestock to eliminate parasites, in the food industry to avoid the presence of insects, in the personal hygiene, and also in antibacterial and environmental programs to eliminate several insects that could be dangerous for the populations and transmit diseases, etc. What are the advantages? Of course, the control of the diseases that are transmitted by vectors. They have been responsible of the almost eradication of typhus, onchocerciasis, malaria, and many other diseases that are transmitted by the insects. And in agriculture, I told you already, to guarantee enough production of food. I don't go, but they have a lot of disadvantages. They are toxic to any living being, including humans. Only 1% of the pesticides reach the target. The other is going everywhere. They, for example, one problem is that they can originate resistance problem. That means that an insect that is continuously fumigated by one insecticide can become resistant to it. Also, one problem is that sometimes if you kill some pests that are concerning agriculture and that produce or destroy many crops, these pests, it's true that are destroyed, but becomes other types of pests that were not able to be there before, but at the end they reach these crops. There are many routes of exposure. One thing is the exposure of the production workers, formulators, farmers, and other thing is the exposure of the final consumer of food. This is the most important exposure route for everybody. The percentage of the populations that work in farms, in industry, etc., is really low. The routes of exposure is the populations, is the intake of food that contain pesticide residue. And also, of course, other route that could be important for the population is the exposure through household insecticides and food preservatives, etc. I have here a kind of attempt to classify pesticides. Classify pesticides is very difficult because we have, I have later on a slide showing it, but now in the world there are about 1,000 substances that can be classified as pesticides. Then pesticides don't have any structural relation among them. There are a few chemical classes, but there are many different structures that can act as a pesticide, since the definition of a pesticide is based in that it's a substances that kill something living. Then, well, I have made the common classification in which we could talk about insecticides that kill insecticides. This is according to the way of action. Herbicides that kill weeds, fungicides that kill fungus, acaricides, molluscicides, rodenticides, etc. And the most well-known group are organochlorides that you have already seen, the organophosphates that are formed by this phosphate ester, the carbamate that has a type of carbamate. But if we are going to the other groups, herbicides, fungicides, as you see, we have many types of chemical structures, like for example, phenoxyherbicides, triacides, imidazolinone, sulfonylureas, aliphatic nitrogen, amide, antibiotics, aromatic compounds, benzimidazoles, benzotriazoles, conazoles, etc., that every one would be a pesticide. I told you there are more than 1,000 active ingredients, and here you have some structures that I just want to show you the characteristics of the structures. This is the DDT. I think that you already know more than well. This is the one organophosphorus. This is chlorpyrifos. You could see the phosphate group. and then how the hydrogens of the phosphates has been substituted by organic compounds, and how sometimes this oxygen from here is substituted by an atom of sulfur, because you put there an atom of sulfur, the effect is stronger. You have here the nitrophenol herbicides, like for example, dinoprop, and you have here also the main structure, the benzenic ring with two nitro groups, nitro. The pyrethroid insecticides, this structure here in the middle would be the pyrethrum, and then the other are substituents to this pyrethrum. Benzimidazole fungicide like albendazole, that is carcinogen, and you could see here the benzimidazole structure, etc., etc. You have here carbamate. This is the carbamate group, which some of the molecules has been substituted by organic compounds, etc., etc. Then, as you could see, structures are very different, and the common descriptor is that they kill living organisms. You have here to give just an idea of what happened in 2004. These data are a little old, but I have to tell you that in the last years, the use of pesticides haven't increased. Since then, we at least has been able to keep the use of pesticides constant. Most important compounds are insecticides, are herbicides, followed by insecticides and fungicides. And we have to say that the pesticides are mostly used in Asia Pacific, North America, and Europe. This is a kind of area where most of the pesticides are used. Then in Latin America, only 14% of the pesticides are used, and in the rest of the world, about 10%. You see here the most important contaminants. And you can see here also how this use of, or how the market of pesticides has increased until 2000. Since 2000, I haven't shown you any data, just because the sales of pesticides has become constant, very small variation, and doesn't deserve to put any more data. You have here the report use of pesticide use per hectare of cropland. You could see that China is one of the countries that has a higher use, also in some parts of South America, small ones. And you should keep in mind one thing is that the parts of this map that you see in gray is not because these parts have doesn't use pesticides, but because there were no data about them. And here you have also several comments about the use in the different countries of Europe. As you could observe, the use is high. I have shown several countries, and one thing that in the graphic that is in the part below the slide, you have detailed how the different countries in Europe the pesticide. And from my point of view, it was curious that not only the south of Europe, where most of the food is produced, use a very high level of pesticide, but countries like Netherlands, Belgium, Germany, France, etc., has also highly used. You have also here see the percentage of use, and there is a movement in reduction of the use of herbicides and to more use of fungicides and bactericides, but small movements, but the use of pesticides has become constant in the last year, not only in Europe, all over the world. The environmental problems of pesticides, I think that has been also shown in this course many times. 90% of the spray insecticide and 90% of the herbicides arrive to a different objective, including non-target species. And these compounds are toxic. Then I'm going just to show you a little the most important toxicity as mechanism of toxicity of these compounds. You have organophosphates, like the organophosphates are characterized because they inhibit the acetylcholinesterase enzyme. The carbamates, then they inhibit the enzyme that degrades acetylcholine, which is a neurotransmitter. These pesticides act increasing the levels of this compound in the human body. The carbamates really produce the same effect, but there is a difference between carbamate. Both of them are able to block the enzyme, but there is a difference in the effect of organophosphates and carbamates, because organophosphate produce a permanent inhibition of the enzyme. Instead, carbamates produce a reversible inhibition of the enzyme. That means that they are less toxic, because if less toxic than the organophosphates. Regarding pyrethroids, they alter the interchange of sodium-potassium, especially in the central nervous system and in the peripheral nervous system. If you look at them, the way of action of pyrethroids are very similar to that of the organochlorine pesticides. And finally, I have put other very common herbicides that are the diquat and paraquat. They are called the ammonium quaternary salts. And these paraquats have a very particular mode of action, because they are able to create a kind of superoxide radicals in the organism, and then they produce radicalized reactions. And the problem with these herbicides is that they need high doses. They are not extremely toxic, but if someone gets poison with them, is very difficult, the treatment, and then many people die because of poisoning with them. I have here some information about pesticides that can be interesting for you. This is just if after course you remember one day I see a course of pesticides and I want to get more information. You can go there to get some external links. Like, for example, the European Union Pesticides Database is very interesting, because you can go there and see the maximum residual limits, and also you have other information about the degradation products, the metabolites, and any other type of information, the residue definition, the recommended doses, etc. The European, in any case, you have the information about the pesticide residue limits in many other databases, and you can get in these links more information. With this, I'm going to move to the last group of compounds that are considered as emerging contaminants. These compounds, I have grouped all of them in the same epitaph, in the same section, because they have a similar chemical behavior. And we are talking about pharmaceuticals, personal care products, and illicit drugs. Let me say that from my point of view, these are the emerging contaminants that better reflect the problem of decrease of population in the world, and also of decrease of the quality of life. Because up to the moment, I have been describing many types of compounds, They have industrial use. They present many advantages in the industry, in the production of several goods. But these are compounds related to our way of living, and they are clearly related in the environment with the presence of big cities. Pharmaceuticals and personal care products are needed to keep humans in good health. Then they form part of our habits, and they are very difficult to eliminate because tell to a person that cannot decrease, for example, his levels of cholesterol because cannot take an agent that reduce the cholesterol. That is something good for health, good for increase the well-being and the quality of life. Then, but the problem is that if you take any pharmaceutical, commonly one part of this pharmaceutical go to the action place and make the beneficial effect for the human being, thus in better physical conditions, but at the end, these pharmaceuticals are also eliminated in part by urine. They finish in the sewage systems. They goes to the wastewater treatment plants if we have it, and if not, they are released directly in the environment. And in the environment, it's true that they are not so persistent like a good organochlorine compound, but they can originate, well, they are continuously released to the environment, and then once in the environment, this compound is also alive. Then that is what happens with these pharmaceuticals, personal care products, and of course here illicit drugs are included. Illicit drugs in an ultimate place, they are pharmaceuticals. They are, of course, banned because their effects on the central nervous system, but many of them have medical applications also. And this has, let me say, thinking, put it here because this is already the end of the class. Everybody is tired, and then I'm trying to make something interesting, not too long, not too short. Well, to see if this time we have take the point. Due to the physicochemical properties of this group of compounds that are high water solubility, low volatility, and often poor degradability, they are able to penetrate through all the stages of filtration, storage, etc., of the stage of cleanup of the wastewater treatment. The low disposal in the wastewater treatment plants has a potential risk to drinking water supply because, well, at the end, if you are releasing contaminants in water, everything is contaminated. One of the problems of these emerging contaminants, I told you, has a high volume of production, high fluxes in the environment, and then, for example, these pharmaceuticals are continuously released. Think in all the people that has a chronic disease and has to intake a pharmaceutical all the mornings, all the afternoons, all the days of his life. And these compounds are different of all other contaminants by a chemical characteristic. And the chemical characteristic is that they don't have to be persistent in the environment, but they can cause negative effect just because they are continuously released. You have here an example of the fate in the environment of these compounds. You see that, I think that they have some animations, but no. In this industry, you see that the pharmaceuticals could be of human use, and then of human use, they reach water through disposal and through excretion by humans. Through disposal, they can go to the domestic waste, and here can go to the landfill, and there they can be leached to the groundwater. Also, they can be, this domestic and hospital waste can be in water, also by human excretion. And then this water could be the sewage and go to the wastewater treatment plant. And in the wastewater treatment plants, we have two products. One is the treated water that has lower levels of these compounds, allow us to recycle water, and then it's very interesting. But you have also a sludge that is able to store many of these contaminants that we are eliminated from water. One of the problems of these wastewater treatment plants that we have nowadays is that we don't know what to do with this sludge. I don't know what happened in other countries, but at least in Europe, where this thing is quite controlled, all are big producers of sludge that we don't know what to do with it. The most common way to eliminate it is to use it as organic amendment in agricultural soils and in other aspects. Then that is something important to know. These sludges are applied in soil. In forest soils is also applied, but mostly in agricultural soils. And these organic amendments can contain these emerging contaminants. Other is the treated water. Up to the moment, we are talking about the wastewater treatment plants are efficient. The treatments are not well. Are efficient for most of the compounds. Many of them are fully eliminated, but there is always a group of pharmaceuticals that cannot be completely eliminated by these treatments. For example, there are several examples that probably you know, but carbamazepine is a kind of antiepileptic highly used, is a very recalcitrant compound that pass over all the treatments of the wastewater treatment plant and finish in the treated water. Same happens with Dicofol, etc. There are other compounds that are in the wastewater treatment plants and that can be eliminated more or less on 90% level, but they are at very high concentrations. Thinking, no, paracetamol. Who of you have take paracetamol quite commonly? Many of us. Ibuprofen, that are the, well, one is are anti-inflammatory compounds highly consumed by the population, and then people, these compounds are at very high levels in the waters that enter the wastewater treatment plant. Even that the treatment eliminate 90% of the concentration of this compound, you have a small concentration that is in the treated water that can be deterred. Is what happens with this. Then surface water become contaminated by these compounds, and these compounds goes also to drinking water. What happens? This is the human use of these compounds, but we have a veterinary use. Veterinary use is also important, but of course, poultry and livestock also consume this type of products. And now the tendency in the world is to go through the very big exploitations, even that there are many people against them just because they contaminate a lot of with the manures that produce. And of course, everywhere has been a rise of aquaculture you need to divide the environmental state of the ocean, and then this veterinary use isn't small, it's very important in the world if you consider not only pets and things that and animals that are at home, but also this industry of poultry, liver, livestock, and aquaculture. All these things contaminate water in the same way as sludges are applied to the soil. Manure is also applying it to the soil. It is a kind of matrix rich in organic matter that makes that improves the productivity of soil and also improves the productivity of the crop. Then have the use, and once in the surface water, these compounds can lixiviate to the groundwater, and then one aspect that is important is their presence or their apparition in drinking water. This is more or less the cycle that these compounds follow in the environment. I hope to be able to introduce you why these compounds are really so important. Well, I have shown you a little the history of these new and emerging contaminants. I have shown you a graphic that is slightly old, but that I like it very much because it's a very good overview, direct overview of the problem. And then really the first time that one pharmaceutical was described in water was in 1970s. During this time, chlorhydric acid was found in wastewater of the USA. It was a very interesting article, but was the only one. During the 18th decade, nobody studied these pharmaceuticals. There were no interest about their presence in the environment. Nobody says nothing. But suddenly in the 90s, start to be considered a problem and a concern. And from then to the present, the number of studies has increased exponentially. Everybody is looking at pharmaceuticals in water, in soils, in sediments, etc., to see what happens with these compounds that are of fully anthropic origin in the environment. In this graphic, you could see several of the most important compounds that are used in the world. You will see that you have the first one are antibiotics. The antibiotics are highly important because few years ago was unclear their effect in the environment. The mention of bacterial resistance appears in few places, but nowadays the World Health Organization considers that the bacterial resistance to the antibiotics that has been created by the presence or the higher presence of these compounds in the environment would become a problem to same people that has get infections, but several pathogens, because antibiotics are becoming efficient for the human. You have here in this graphic also one aspect that should be kept in mind when we talk about these compounds in the environment is their metabolization. You see that you have in this graphic, those bars that are in red means the amount of pharmaceutical that is excreted and changed. That means as the pharmaceutical. And you could see that there is a very big percentage of the pharmaceutical that is always excreted at this pharmaceutical. Then in green, all these, sorry, I make a mistake. I'm already finished this. I have been trying to talk so many times. Then the red bar means the amount of pharmaceutical consumed. The green one is the pharmaceutical that is screened and changed. But as you see, you have some yellow and blank bars, and this means the part that are screened as phase 1 metabolites and phase 2 metabolites of this compound. Phase 1 metabolites, for those people who cannot know it, are those that are produced in the human organism by basic reactions, as for example, oxidation, hydrolysis, etc., etc. And phase 2 metabolites are those in which the pharmaceutical don't change their chemical form, but is conjugated with one molecule that makes them more polar in order to eliminate them by unit. Then, as you could see, this is a very difficult terrain for an analysis because you have the unchanged compounds, the formation of complex, like complex with glucuronides, complex with sulfides, etc., etc. And finally, you have other metabolites that could be oxidized, hydrolyzed, they cannot. The most common are the processes of oxidation, that mean hydroxylation and oxidation, but you can have methylation, etc., etc., and other processes. Then this is a world of many molecules that we should study. They can be dangerous for the aquatic biota because they are designed to have a physiological effect. Pharmaceuticals could be of natural origin and could be just of synthesis. They has been think to act with any of our enzymatic systems, any of our cycles, anything in order to improve our health. They can improve the health of the human being and be damaged for a fish. Then, but they are designed to go to a living body and to have an action on it. And also you should keep in mind that all this array of metabolites that are described first are highly soluble in water than the parent compound, and could be more dangerous than the parent compounds because nobody knows too much about the physiological effect of the metabolites. They has been considered up to now not too important, and well, that is a problem. I have shown here a very few slides just to show you a little the metabolism of the pharmaceuticals in the human body. I see that this slide is in Spanish, sorry, but sometimes for me it's difficult to remember that I have to change, but I told you the technical language is very similar, Spanish and English, in many languages. Then we are talking about pharmaceuticals, the different reactions of oxidation, hydroxylation, the alkylation, desamination, hydrolysis, etc. And you could see this is the salicylic acid. The salicylic acid is a metabolite of something so common like aspirin, and you could see like this aspirin could be degraded just by oxidation, in which this ethyl acetyl group is missing and is transformed in the hydroxide. This is the salicylic acid, and then also this could be conjugated with a glucuronic acid in order to eliminate, as to show you. Here you have the case of the imipramine. The imipramine has a most complex metabolism with the methylation and the alkylation, and then once has been the methylate and the alkylate by hydrolysis, then is hydroxylate. And you see that the addition of oxygen groups is always a way to make a molecule more polar, and then it's important. You have also conjugation, and of course they are described by unit. You have here the percentage of each one of the metabolites, because it's very important the percentage. Not all the metabolites are in the same amount, and not all of them are so representative of the consumption of these compounds. And this one is the metabolism of the ibuprofen. You see, like it follows also a way to become hydroxylate one side, and also they are combined in a phase 2 metabolism. And then all these metabolites can be from water. In fact, we have been performed several studies with the ibuprofen and the presence of the hydroxy metabolites, and the oxide are frequent in water. And with this, I finish with the pharmaceuticals, and I would like to devote the last few minutes of this class to talk about the new or the most emerging contaminants in our environment, that are microplastics. Microplastics are a real problem. I'm going to be brief because I'm going to keep the original time schedule, and I see that I have about 10 minutes to talk about them. I think that is enough because microplastics are slightly different to other contaminants, and is the newest emerging contaminant that has appeared recently. And in my opinion, it is one of the most important because of the persistence. There are some facts that, let me say, reflect the problem with these contaminants. I have shown a few of them in this slide. Is the growing plastic production. You see, in 1915, 1.5 millions of tons of plastic per year were produced. In 2006, this production was increased to 245 million tons per year. Nowadays, the production is already now, and it's about 300 million. Then this produce many plastics. What happens with the plastics? That they are highly stable, they are fossil fuel derivatives, and they are synthetic polymers, has an important persistence. They are very difficult to degrade. They are very light. Then, for example, for the transport of the products, are much better than glass because they become cheaper. But the problem is that the largest part of these plastics is still dump. Well, the landfills, in the water, the wind takes out, beds. Then commonly this plastic finish in the oceans. Then maybe we can make us a question how much plastic are in the oceans. It's a worldwide problem because maybe once countries threw it more plastics to the sea than others, but at the end, all the oceans are connected. Then you could see here, these are the oceans, and you could see this is a satellite photo that shows what happens in the world. Oceans are interconnected. In fact, they have some characteristic currents that makes them what they are. And you see that the oceans have microplastics. These yellow points are not exactly only plastics, but are the waste is accumulated in the ocean. That is a shame for the human being because it's incredible that we don't take more care to preserve our environment. You see, this is the same photo, but taken with other filters, etc., and you could see that appear some places where these plastics are accumulated, just because of the different currents. And you see, this gives an idea of the intensity of the accumulation, and you could see, like, for example, here near of Australia, in this part near of the United States, also South America, is becoming a very, very big landfill of plastics and other types of waste. Here, these photos, I think that aren't selective only for microplastic, only for plastics, but plastics are the most, let me say, plastic is the most important waste in the seas because other types of materials are degradable. A piece of wood that was thrown away to the sea 200 years or 100 years ago has already degraded, doesn't left any residue. One piece of plastic that could already be thrown away 100 years ago is still in the sea, almost. Then that is the difference among one and another. What are the microplastics? Well, we have this amount of plastics in the sea. It's true that plastics are very little degradable, but once they has been 100, not so big, 50 years in the sea, the mechanical forces and the UV action, etc., are able to destroy small particles and they become microplastics. These microplastics are small. How much small? Well, there is not a established definition of microplastics. Nowadays, there are a very important discussion about this. What is a microplastic? What is a nanoplastic? When it becomes a macroplastic, but the commonly accepted definition is that a microplastic is any piece of plastics below half millimeter. Below 5 millimeters. Then this is the limit that always has been used to define microplastics. First words were more devoted to the biggest one. Now we are commonly studies are focus on microplastics that can have 20 microns, etc. Plastics below 1 micrometer are now called nanoplastics, but the methods to determine nanoplastics are still under development. They are not easy to determine, and then we don't have really an important, a very big knowledge about these microplastics. The problem of microplastics was raised by Thomson et al. In 2004. They published in Nature an article called Lost at the Sea, where is all the plastic. And this was like the beginning of the study of microplastics. And nowadays, microplastics has been detected in sediment, water, biota, food, etc. Microplastics has been found everywhere, but the main problem in the research is that there is no agreement on concentration. Here you have a slide where you could see the microplastic particles that has been identified in the plankton, in a graphic you could see like they do look like these are 50 microns, and this I don't know what is this, but probably is an electron microscopy imaging, and here you have the amount of microplastics that has been observed in the plant tau, and also is it compared with the production of microplastics. Then as the production increases, the amount of microplastics that are in water increase. Then you have several examples of how they has been counted in several stories. There are many words about microplastics. Nowadays, everybody is determined microplastic. And here you have how they are identified in the gut cavity of the Mytilus edulis masses. You could see here yes, here these are the microplastics, and how they can be accumulate in the organism. Well, regarding the microplastics, I don't go to mention any more things. In addition to these plastics, to these microplastics that I mentioned before, and that they are coming from the UV sources and the mechanical action of the waves. There are also primary microplastics that are synthesized already as microplastic, that they are used, for example, in the defoliant creams and many other things. We have filled the world with microplastics, and important thing is that we talk about microplastics as we talk about pesticides, but there are not only one microplastics, there are many types of plastics: polyethylene, polypropylene, polyester. Then if we are just talking about polyethylene, you can have low density. There are many of these compounds, and they can have a toxic effect. We are still discussing whether microplastics are toxic or not. They shouldn't be here if from the moment that they are able to accumulate in a muscle or to accumulate in a fish, of course that they haven't to be in the environment. Then it is important to present. Just to finish, I have shown you the decision, the European legislation about prioritary substances that are, I back to my first about the persistent organic pollutants, just to show you what are those that are legislated, and then all the others could be considered emerging contaminants. I have shown these ones because from my point of view, I think that they could be the most interesting and that they are maybe the most dangerous for the environment, but there are many more. You see, this is the second part of this European legislation. These are the compounds that are considered as emerging one and are now being introduced in the legislation. And in the moment that one of the compounds become in this list, it left to be an emerging contaminant and will become a traditional one. Then nowadays, many of the emerging contaminants, or well, many, no, few of them are included in this list, but there are still many that are not legislated, the toxicity of which is not fully demonstrated, even that we have a lot of data that make us think that they are really toxic for the environment. And this problem of emerging contaminants clearly deserves to be studied if we want to have a better world for us and for our destiny. I have finished here this presentation. I would like to ask if there is any question. Any question for the public? From the public? Nobody have a question? Well, then today I'm going to ask you for 5 minutes break, just to drink a little of water, to see a couple of things, and I will return exactly in 5 minutes to give you a short tutorial about the handling of these emerging contaminants in water. My idea is this tutorial will be follow today and tomorrow, and then today I will talk to you a little bit about how to sample water, and depending on the type of water, and how to extract and isolate these contaminants. And in the next tutorial, we will talk about how to determine them by liquid chromatography mass spectrometry. Mostly, I will show slightly maybe any other technique, but the word is let me say, block with this technique because it has many advantages. Then, well, I close here. My clock is 11. That must be there about 4 in the afternoon. And then I will back in 5 minutes. Okay, thank you. Not this one. Un moment. Well, then I'm going to start without any more delay. Welcome everybody to this second tutorial, handling an instrumental characterization of contaminants of emerging concern in water, and I'm going to talk about the analysis of these compounds. The determination of these compounds in the environment have several complications because they are at very low concentration in the environment. You have here this slide that shows how these methods are. Then you could see here that the analytical methods have several aspects, sampling, extraction, and cleanup, and that once the sample has been cleaned up, there is separation, identification, and quantification. This is like any other analytical method. To say more or less all of them have the same steps. One moment, because I don't know what has been done, but this isn't working for me. Regarding how much time is needed for each step, well, this is a long step. Sometimes many of these methods require more than 1 day to be performed, and you have here several aspects. Sampling, you see, sample preparation has traditionally been called as the bottleneck of the process because it's long, tedious. Many times you need to do a lot of cleanup, then analysis, and then data treatment. I have to tell that data treatment is now getting even more important than sample treatment, because you know that with the new systems of accurate mass spectrometry, you can get a lot of information, but the problem to get a lot of information with a technique is that then you need time to process, to study, to establish, etc., and then it's improved. Then sampling is not, I'm going to start by the sampling. And first thing that I have to say is the sampling is not the longest step, but is a very important one because it's responsible of the homogeneity of the sample, and you should know what are you doing and what are the information that you will get with the sampling that you do. I don't know what happens today with my screen. I hope. Let me unshare again in order to see if I can pass these presentations as I'm talking, because for me it's much more comfortable and I get more concentrated in the class. But yes, well, exactly. Then regarding the sampling, what we look for is what we find, because it depends on what we are looking for, how we took the sample. Then sampling is a critical part of the analytical methods, and if it's not performed using the appropriate techniques and according to specific objectives, the analytical result might be highly biased. The sampling strategy should reflect the composition of the environmental compartment. In addition, the sample should be retained in order to determine the exact concentration, and the design of the sampling plan is linked to the effects of sampling and to the specific information that we want to collect. It's not the same to take a sample to determine volatile compounds to take a sample to determine polar compounds, because in the case, for example, of the volatiles, you should keep all the precautions to avoid that these compounds get disappear of the sample before the analysis. Then one thing very important when you are going to sampling and to establish a sampling strategy is to follow carefully the steps. The sampling strategy should reflect the composition of the environmental compartment. In addition, the sample should be routined in order to determine the exact concentration. And one thing is that the design of a sampling plan is highly important. The sampling plan should be designed and should contain also information about the storage and preservation, transport, stability, and sometimes the analysis. Okay. Then, when you establish a plan, you should take into account a series of considerations, like which contaminants are to be analyzed. That means how they should be preserved. If the samples are for qualitative or quantitative analysis, if the contamination that we are looking for are organic or inorganic, or run also the type of compounds, because, for example, the contamination could be random, uniform, irregular, stratified, or following a gradient. Then many times some preliminary tests should be done and should be recommended. Also, we have to look at what are the requirements of the analytical methods to be applied to the samples. Then also we have to take a series of decisions, like for example, the selection of the sampling points, the sampling frequency, the sampling size, mass and volume required, the number of sampling units at each sampling site, the selection of sampling devices, which is important because we have to establish container and cleanness requirements, the sampling type, if it is discrete, composite, multi-level, or maybe passive. We will talk slightly about all these decisions that you have to take during the preparation of a sampling plan. And also you should, for example, take a look to the sampling site records. You should know the UTM coordinates in order to go always to the same place, to have a map, and to find the description of the access, because things looks very easy sometimes when you make a plan in the map, but when you go for the sample, could be different. You have to take a decision about the dates, and you should keep in mind the weather conditions, because, for example, you can take a sample, but if it's raining, sometimes it's better to wait a little. Also, you have to prepare the necessary material, and you should keep in mind that the first time that you go to a place for a sample is always more difficult, because you have to find exactly the place. Sometimes you don't know the roads or the location. Sometimes you go to a location and find that it wasn't what you expect, and then you have to look for a new one. But once you have established this plan and you have a record of data, things become easy and the sampling become harmonized. You have seen information that you could take, like for example, some photos are of hell. Now with the Google Maps, you have a lot of work done, but you still have to use these coordinates. Then you can take several types of samples, depending on the type of contamination that you are studying. For example, you have critic. When there is a critic sample is when you, for example, are near of punctual source of contamination. And then, for example, if you have an industry and you want to see if this industry is producing a kind of contaminants, first sample would be ideally near of the industry to see if this contaminant is present or not. You can't have interest in make a systematic sample, for example, because you want to see how long this contaminant appear in the environment, and then you have to take a map and to make a systematic sampling in an area. Then the ideal thing is like is showing this slide, and you have put lines and have quadriculate the area. And finally, you have also the possibility to make just a sample to see if a contaminant is present or no, like for example, a random sample, in which you take several samples along a distance to see what happens, if there is a contaminant or no. In a few samples, if appears, etc. Then these are the types of samples that you can do. And also there are several types of samples that you can collect. For example, in this slide, you could see you have here a scheme of the different types of samples that you can take. Here I have in this presentation, I have restrict to water samples just to don't make it too long and too tired and too heavy for you, but in this scheme I have also how to take soil and sediment samples, and it's interesting because many times you are looking for water, but you are also looking at the other environmental compartments, just because they are in contact and they have also contamination. Then the grab sample is just a punctual sample that is collected at a given point in a short period of time. Has advantages because commonly grab samples are easy to take. They represent the compartment at the exact time of the sampling. And this type of sampling is very easy and is recommended when the source remains constant. But this happens little times in the environment. Commonly fails to detect contaminations events coming from a point source, and the surface or deep is determined by the current. Commonly, you know that the rules establish that in the case of the water, you should take the grab sample in the middle of the stream, in the middle of the wide of the stream, at a deep of about 20-30 centimeters. But this depends always on the deep, depends on the intensity of the current, and depends on the circumstances. You have here marked the different types of grab samples that you can. Then there are other types of samples, like for example, mixed samples. And then the sample is collected and mixed in the same, several samples are taken in the place and then they are mixed in the same container. This can be more different depending on the sampling sites, but one of the advantages of this type of samples is that give an average of the compartment characteristics. I mean, if you, instead to take in a river one sample of water, take 5, 20, 25 and make a composite sample, the sample will represent the average of the samples. Then these mixed samples are very useful for long-term monitoring. And for example, they are used in many works that make that monitor wastewater treatment plants. Commonly, water treatment plants take 24 hours composite samples. That means that they have an automatic matching that every hour takes about 200 milliliters of the sample, and then they make a composite sample with these 250 milliliter samples, and then it gives a representative of the composition of the wastewater along the 24 hours of the day. We can also have some interest in know what is the difference in the environmental compartments during the 24 hours, along the deep of the compartment, and then in this case we take multi-level samples. The taking of the multi-level samples are samples that are located, that are taken at different places, but instead to be in different places in the area, are in different places in a vertical line. That is very important if we want to know if there are differences in the sample with the dip. Then the characteristics and, in fact, the characteristics and the composition of an environmental compartment can vary with this vertical dip. This is very useful for tracking, for example, contamination plumes in groundwater system. Then you see there are special points that are able to that are special devices that are able to go in deep and then take in this deep different samples. Much more easy in the case of the soil and in the sediments that you take just cores. Cores are empty cylinders of some deep, 1 meter, 2 meters, 100 meters, and then you could introduce them into the sediment and take it, and then once you have taken the sample, you can cut it in different proportions. We have nowadays other types of samples that are samples called passive samples. These passive samples have advantages, as for example, they are collect a sample over the time. This is a quite updated sampling strategy, and commonly they are devices that contain a solvent. They are put into an environmental place for about 15, 21 days, 1 month, depending on the type of sample that you use. And then you take the sample, elute it, and obtain the analyze that has been in this water for all this period of time. These passive samples have the advantage that combine sampling, analyte, isolation, and pre-concentration in only one step. Then this is a way to collect or to sampling a body of water during a long period of time with it have too many samples or too many gram samples that sometimes are difficult to analyze. I have shown you this sample system. All of them has advantages and disadvantages, but one of the advantages of this passive samples is that they can detect punctual release of compounds, because sometimes if an industry make a release of toxic compounds in the night and you go at the next morning to take samples, you cannot detect the compounds. But with this type of devices, let me say that they are able to retain a concentration of the compound that represents an average of that of the whole day, whole month, etc. That are the different ways to sample, and then it's important to keep in mind the storage, preservation, and transport of the sample. Important thing during storage, preservation, and transport is that the composition of the sample must be preserved, must be the same that you took in the river sugar right at the laboratory. Then what are the things that can change the composition of this sample? You have in the consideration several aspects. For example, biological action. Samples of river samples are not sterile. They have a lot of microorganisms that love the and that can degrade compounds. Also, hydrolysis could be a physical process that could follow in a bottle, then it should be stopped. Also, if, for example, a sample, an analyte is highly volatile and you took only half of the bottle that you have, the analyte can be volatilized. In this case, you should fill up to the end the bottle. Also, should take into account the material of the bottle because the contaminant could be absorbed to it, and also you have to prevent photodegradation. That is the reason why most of the bottles that you use to take sample are in amber, dark, or of a color that don't allow to pass light through them. What do you do commonly when you took a sample? You make a pH control. You could add several chemicals, for example, some antibacterials to inhibit the bacterial growth, or you could add antioxidants to avoid oxidation of the analyte. Then you have also light preservation, which is amber material, and very important in order to reduce any type of reaction is to keep the sample cold, the refrigeration of freezing. Regarding the refrigeration and freezing of the water samples during the transport, I have to say that you should keep in mind that all these recommendations are highly useful. It's very interesting to try to preserve completely the sample, but that you should go for a sample and be able to take it to the laboratory. Then you should keep in mind rationality and conditions. Then, for example, during a time, everybody says that it was highly important to freeze the samples. In our opinion, it's better to refrigerate them because many times when they arrive to the laboratory, before to frozen or to keep it for a while, is needed to filter or to make any process, and then it's better that the samples arrive not frozen to the laboratory, but they could be the most easy way to preserve pulse or samples is to take some refrigerators that goes with ice, and then you could keep easily the samples at 4 temperature. And then you also could mail the samples to the laboratory depending on what happened, if you have to move the samples between laboratories, etc. Then you should keep in mind all these considerations and all these actions. I don't want to be extremely defined with them because they sometimes depend on the compounds, on the chemical class, on the matrix, and the analytical technique, and of course, on your resources. Then one thing is what is ideal, one thing is what you can reach, but you should keep, you should know all these things in order to try to do the sampling as best as possible. Bueno, regarding the sampling devices, depends on the compounds or the chemical class to be analyzed, the matrix, and the analytical technique. Sampling devices and containers have to be chosen many times depending on the compounds to be analyzed to avoid absorption or contamination. For example, if you want to determine organic compounds, I don't know, for example, polychlorinated biphenyls, pharmaceuticals, etc. Sometimes you should avoid plastics as they contain, for example, phthalates, and and this is something that you should keep in mind. Sometimes glass is not the best option, not because many organic compounds can be absorbed on the glass. For example, this happens with all the persistent organic pollutants. They are so insoluble in water, and commonly they are absorbed in the glass, but it happens also with some pharmaceuticals, etc. Then, in the case of the organic compounds, you should look very careful at what type of bottles you use. Then the best options are the borosilicate amber glass with the problems during the transport that they can break. Also, stainless steel material is quite useful because the stainless steel mostly gives metals, and then organic compounds are not so absorbed to it. But if you are in an area where the water is quite acidic, you must be also careful. Teflon, Teflon could be a very inert material, but you should avoid it if you want to deter perfluoralkyl substances. And in the case of polyethylene terephthalate plastic, you can use them for sampling, but you should keep in mind that you can't reuse the bottles. Then, in the case of metal, you should avoid glass. Metals are nowadays ideally sampling in plastic. At the beginning, they were sampling in a material that was clean with nitric acid, etc., but nowadays there are perfect plastics that helps to take these samples. From volatile organic compounds, I already tell you, don't leave headspace. And one important thing with all this material that you use to determine the sample is that you should avoid, that you should establish a cleaning protocol, because the material that you are using in the field must be perfectly clean in order to avoid contamination. Commonly, the protocol involves, in the case of the glass bottles, to burn them at some temperature, then to clean them with solvents and to evaporate the solvent, then to clean it with distilled water in the lab, and then once you are in the field, you commonly take first a small volume of the natural water that you want to take, you clean the water with it, and then you take the sample. Commonly, if you want to make an interesting sampling and you want to ensure that don't have any field contamination, one important thing is to take field blanks. The field blank, what are the field blanks? Well, it depends. You should decide how many field blanks, what field blanks, and in the case of other matrices, they are not so easy because you never know what can find in a sample. But for example, in the case of water, it's very easy. You just fill one of the bottles with water and then close it and takes with you along all the travel just to see if it has any contaminants. This is one type of fill. Other type of fill blank is just to keep with you a bottle with water, and once in the field, pass to one of the sample bottles and take a sample in order to see if there is any contaminants that should be erased from your result. Well, I think that these presentations go today slightly slow. I will try to, well, I don't know what happens. Everybody is hearing me well? Then, regarding the types of water samples, you can take, for example, groundwater. Normally, in all cases, when you take a sample, you measure a series of parameters that are very easy to determine, that gives us an idea of the quality of the sample, because sometimes the mineral content, the organic matter content, etc., could interfere in the analysis. You have normally you determine dissolved organic carbon, chemical oxygen demand, pH, temperature, conductivity, and oxygen dissolved. The groundwater samples are obtained from wells or spring. This is complicated, especially in the case of groundwaters, because these wells or springs are not always available in the best places, or areas not available. but it's the only way that we have to establish the movement of the contaminants. You can also take interstitial water, that is good water trapped in the ground, with sampling pumps through filters or through passive samples. And these samples, when arrive to the laboratory, they are going to be analyzed within 15 days. Commonly, I recommend just to storage at 4 degrees centigrade. Depends slightly this recommendation on what are you determine, but with this is enough. The most common sampling is that of surface waters, that is performed in rivers, reservoirs, lakes, etc. Sampling could be done with ships from the bridges that are a way to be in the middle of the stream, in coastal platforms if you are going to the sea, upstream or downstream of the agricultural, industrial areas, urban discharges, lake outlets, tank wastewater treatment plants. You should study your, I don't know, depending on the types of surface water, but normally you have to study the basin in order to see what are the potential points of contamination. And if you see a potential point of contamination, you can establish a sampling point before and after. This is more for the sampling plan than for this, but I think that is interesting to mention it. Water, it's easy to test, as we have directly, wastewater is easy to test because commonly the wastewater treatment plant has the facilities to do it. For example, we here in Spain all the years sample a couple of times the wastewater treatment plant of Valencia, and then they have all the systems. We just go with the bottles and took the sample, and it's marvelous for us, but sometimes you have to do it. And many times the sample is slightly complicated. In the case of wastewater, the most common way to sample is these 24 hours composite samples that I mentioned you before. You have here a slide showing you the most important sample devices. Then you have drugs of this type. This type of grab samples and drugs are very useful because you have to submerge them in the middle of the stream, and then once the bottle is covered by water, you open the tap, and then the bottle is filled with water. This is a homemade drug that we had, but you can also buy several commercial devices that are quite useful. This has the advantage that we can use glass bottles because for the type of contaminants that we were determining at that moment, it was interesting for us. This type of sampling devices provide discrete or composite samples, provide rapid descent into water, prevent vertical contamination, have a proper closing mechanism, must be easy to use and maintain, are of appropriate capacity for the samples, can be used for surface and groundwater, used for shore platforms, boats and brides, etc. And then you have also the cavity coder that can sample the water for over 100 meters. These cavity coders are commonly special devices when you have to sample very deep current of waters, and then you have to go very deep because in this moment the pressure don't allow to introduce one of the previous systems. And then other devices are pumps. Pumps commonly helps. You have here the scheme of one of these pumps that are able to take a water sample. The pumps must be parts prior or of the sampling to avoid any sampling stagnant water, because otherwise you can have a lot of microorganisms or other or accumulation of metals. If, for example, you have a pump like that of the photo, there is a very wide variety of pumps, and the choice will depend mainly on the distance to the sampling point and the facilities of the location. Normally, these pumps are used for groundwater, interstitial water, but also can be used for surface water, especially if the surface water, the stream runs deep among gorges or something like that. Then other types of water samples are the waters that are sampled in multi-level devices. Then you have here how they are taken. These multi-level devices are very useful if we are taking samples in multi-level wells that are built for the occasion and that can be unique in terms of materials, dips, or pumping systems. Commonly, you need one of these multi-level systems. There are some of them commercial, but you will need to customize to the needs of the well that you want to sample. In this case of these multi-levels, as you could see, it's just a sample that allows to take water independently at different depths, and the materials to construct these samples must be chemically inert. These samples are used for groundwater and are very specific. And then finally, to finish with the sample, and here, and I left a little comments that I would like. Can you hear me well? I ask you because my computer is advising me that my internet connection is quite unstable.
No, we can hear you, no problem.
Okay, it's just in my case. Well, these are sampling devices that I would like to know, to mention, because they are becoming highly important, that are the passive samples. The passive samples, as I have mentioned before, are samples that are introduced in the water, and then they are left there for a few time, from few months, normally 28 days. Then these passive samples are solid devices that are based on the free flow of analyte molecules from the sample medium to a collection medium that is able to concentrate them because the different affinity. Then the deployment of these devices is important, and one important aspect is that you should calibrate these devices. Now, in fact, its use is not as general as it could be, because the calibration of these devices is very complicated. Normally, if you want to calibrate it, you have to use any kind of contaminant gradient over a period of days to calibrate. Commonly, it's applied to surface water for integrated time detection, or for sites where it isn't easy to collect or pump the water. And nowadays there are a variety of passive samples that are commercially available. Then you have here some examples of the most highly used. The passive samples devices are always based on sample diffusion. They could be SEM catchers, that they all membrane samples that are the most popular, and you have within the membrane samples semi-permeable membranes device that are most effective for nonpolar compounds. In fact, they are considered as mimic of the concentration of biological analyzing biological fatty tissues. Then there are also passive in situ concentrations, some concentration or extraction samples like POCIs. These POCIs are, I think that nowadays the highly used device, because they are highly appropriate to retain polar compounds, like could be the pharmaceuticals, it could be the pesticides. Then there are also support liquid membranes devices. The support liquid membranes devices has the inconvenience that they are really more unstable than the ones, solvent devices and solvent filled devices. There is a class, you have here several photos to illustrate these determinations. Then you have also different apparatus, etc., etc. You have here, for example, solid phase membrane devices that consist of solid of a sealed tubular low density polyethylene membrane. Commonly, this polyethylene membrane has between 25 to 250 micrometers of thickness, and this is filled with a lipid of high molecular weight, typically thiolanes, and then commonly the contaminants pass through this low density porous polyethylene and are retained in this liquid that is highly. Then once you have these devices for 15 days in the water, you took them to the laboratory, and then it's important to extract the analytes. Then you have here several things that you can do. For example, in some cases they are washed with sodium hydroxide. They could be dialyzed versus non-polar solvent, cleaning band sizes, pollution, chromatography, etc. And then the analytes are determined. But this is not always like that. Sometimes the illusion is much more simple. For example, with hypothesis, you just have to put the device in methanol. And these sample devices could be used for matrices as surface water, groundwater, agricultural runoff, industry effluent, and commonly they are specially designed to deter pollutants that are highly non-polar, like polycyclic aromatic hydrocarbons, at least in this case, PCBs, dioxins, furans, organochlorine, pesticides, organotin compounds, chlorophenol, polar, some others are for polar biocides, etc. Then I think that, well, this has been some show of how sediment samples are taken, just as the last slide, to show you that we can extend even much more the type of matrices that are determined in water. And then, well, I don't know if do you want that I follow with this class longer, or you would prefer to make a few questions that you have for today and we close today the session. Let me know what you will like to do.
Yes, participants.
Do you have any question in this moment that I have finished the water sampling? If you don't want to make the question, or you have problems with your audio or anything, you can just in the chat.
Yes, participants, if you have any questions, please.
We can finish in 5-10 minutes. Or...
Any participant, if you have any query, please raise your hand.
उसके बाद DST और MSc है,
If we can finish in 5-10 minutes would be better. Hello, Dr. Gulshan.
Yes, sir.
Hi.
If we can finish in 5-10 minutes.
Sure,
And-
definitely. If I'm just wondering if any participant is having any question, then that can be raised. Otherwise,
Well, in this case, I can follow a little with instrumental determination if you wish, or I can finish, depending on you.
Yes.
Yes, means finish. Well, I'm going to, I don't know what to do. Follow?
there is no query from any participant, Yolanda.
Then I will follow a little with the determination of analytical contaminants, and I will finish this part. Is it okay?
Yeah, it's okay.
Okay. Then the next step is, once the sample is in the laboratory, we have to extract the matrix. The extraction is very important, always has been considered as the bottleneck of the extraction procedure. And then you have here a short scheme of how matrix are analyzed. In this first scheme, then I'm going to keep in all the moments close to the, in any moment close to the water analysis, but the samples in the environment could be much complexes, and then you can have liquid matrices, like for example water or some biological samples, and solid matrices. In the case of liquid matrices, of course, the most common methods are the liquid-liquid extraction and the solid phase extraction, and they are the two methods to determine the answer. Nowadays, and because we are also entering in issues about the green chemistry, one important step is to make safer for the environment the method that we use. And then in this case, we have microextraction processes that has been developed. But these microextraction processes, I advance to you that they are based on liquid-liquid extraction and solid phase extraction. Then, in the case of the solid matrices, you have to do previous steps before a liquid-liquid extraction, of course, or a solid liquids and solid phase extraction, that is an extraction with commonly an organic solvent. You have source length liquid extraction. You can use other things like supercritical fluid extraction, microwave assisted extraction, and pressurized liquid extraction, and matrix solid phase dispersion. With this, I want to say that you can use almost the same method that you use for water samples to clean the extracts of the solid sample, just to give you an idea of how you can apply this analysis. The liquid-liquid extraction, make sure that you know, but I have put a few photos just to show how it is to extract directly with an organic solvent immiscible with water. That could be a quite polar non-miscible organic solvent like ethyl acetate or dichloromethane that gives a wider range of the polarity of organic compounds, and then also can extract hexane, petroleum ether, or other lipophilic compounds. Then regarding the factors that determine analyte recoveries, you should keep in mind the affinity of the analyte for the solvent, the total volume that you are using to extract the sample, the number of extraction stage, the pH of the water, etc., etc. You have here this that show you how to make the extraction. Commonly, you put in the water and the solvent in a phenyl and agitate. Keep in mind that it's very important to to open the device time to time in order to eliminate gases that could be formed. But this method has many, many disadvantages, like intensive work. Yeah, by sure that if any of you has been working with this, you know how much time consuming is, and it's tired because you don't have any automatic agitator. You have to do it with your hand. And then also it's important the extensive use of glass materials, the high volume of high purity solvents, the emulsion problems, or the pretreatment of the samples when the sample is other liquid that is water. Then most of the waters are nowadays analyzed by solid phase extraction. Solid phase extraction is not a new technique. It was developed in the 17th decade and involves the solid phase extraction, the isolation and pre-concentration of the organic compounds from liquid matrices by passing the sample through a short column that is filled with a sorbent. Then, once the sample has passed, the analytes that are isolated or retained in the solvent are eliminated by a small amount of an organic solvent. Then it is easily incorporated into automatic procedures and also can be easily incorporated in online systems, and solid phase extraction has become nowadays the main technique. Why? Because it has some advantages. It's rapid, it's more reproducible, avoid emulsions, and provide low sample volume. Then, what are the solid phase that I am able to retain the analytes in this process? Well, there are many solid phases nowadays and many types of them. And you have here some of the most highly used. Commonly, they have several abbreviation. I have to say that depending on, you have, for example, C18, that is the traditional octaethyl silica, SDBD, that is a highly cross-linked styrene divinyl benzene polymer, or it's also called PRP1, ECB, that is graphitic GBC, sorry, that is graphitic ballast carbon, is a mistake in the slide, that is also known as carbopap, carbograph, etc. Also, you have environ P, styrene divinyl benzene copolymer, Licrolute, that is a reverse phase, C18 phase, and also an Oasis HLB. The Oasis HLB is a commercial name. The phase is HLB, and this means that it's a phase that has an aerophilic lipophilic balance. This aerophilic lipophilic balance is obtained with a poly-divinyl benzene co-N-vinyl pyrrolidone. Then you have here, I have said, this is a commercial, I have the three names of the commercial phases, the Strata X, the Water Oasis, and the Supelco, but I have using this, let me say, commercial advertisement because I consider it very clear about the identity of this aerophilic lipophilic phases. Then you have here how the divinyl benzene polymer is the lipophilic part of the phase, and this pyrrolidone is the hydrophobic part of the phase that is able to retain quite well the polar phases. These are water-wettable phases, and has a very good characteristics because, at least in theory, they are stable between a range of pH between 0 and 14. And these phases has, in addition, the advantage that they don't have these silanol interactions that sometimes retain few compounds. Then this is in the case of the Oasis phases that are an aerophilic lipophilic balance, and nowadays have become the most used phases because they are sometimes not so nice like other chemical, let me say, chemical devices that you can use, but let me say that they are highly robust and they retain many of the highly polar compounds. Nowadays, there has been a change of the contaminants from these traditional persistent organic contaminants that all of them were highly non-polar to the polar ones that really are worse retained in some of the phases. And then most of the research regarding the solvents that are used for retaining these compounds are focused on the different phases, the different solvents that you use. Then you have these hydrophilic lipophilic balance phases or solvents has given pass to other ones that has been designed especially to retain better polar compounds that are the mixed mode. What means the mixed mode? The mixed modes mean that you are used one of these hydrophilic lipophilic balance phases combined with some ionic interchanges. You know that in ionic interchanges can be strong cationic interchanges, weak cationic interchanges, strong anionic interchanges, or weak anionic interchanges. You have here these, let me say, derived phases, and you have here this structure of HLB, and then you have the different functional groups that act as ionic interchanges. These are for cations, then you use, in the case of the strong ones, sulfate groups, and in the case of the weak ones, these weak acids, and the same happens with the anions. In this case, you use quaternary amines or secondary amines. Then this has, let me say, make a wall this part of the solid phase extraction, but in order to look for, let me say, a kind of universal method that would be able to extract almost all the compounds that can be present in a water sample. You have here the solid phase extraction, different parts of the extraction, and then this is a proposal. This proposal is from one of the commercial trademarks that produce these phases, but it's interesting and show you how just using 2 different phases and 4 analytical methods, the idea is to extract almost any contaminant that could be in water. In addition to these common solid phase extractions, you have the selective one, and in the case of the selective one, by sure there are 2 types. One are the immunoaffinity columns that are based on the interactions antigen antibodies, and also molecular imprinted polymers, that the molecular imprinted polymers are based, have a more interesting chemical design in which, let me say that the molecule is used as template to create a polymer through it, and then once the molecule is eliminated from the phase, you have holes that has the exact form of the molecule. In this case of the molecular imprinted polymers, the most complicated step is how to eliminate this template, or how to use a template slightly different from the molecule that could be distinguished in order to know what is the amount of analyte. But in both cases, there are some selective solid phase extraction that can be highly useful for particular analytes. And just as the last part of this water extraction, and then I'm going to finish now, I will try to be brief, don't be too longer, is that you have a number of microextraction procedures to eliminate to extract waters. But sure that some of them are already well known, that are the solid phase microextraction. The solid phase microextraction, you know, that is to reduce the the solid, the amount of solid sorbent, and then you have small fibers, small bars like sortive bars, or even thin layer microstructures that are little used, but the common ones are microfibers and sortive bars. Then you have also cases in which the sample flows through the solid phase, and then we can talk about in tube solid phase microstructure, in needle, in tip, etc. And then you have also the liquid phase microstructure, like single drop microstructure, dispersive liquid microstructure, or assisted membrane that are very common. Then here you have some examples of the solid phase microstructure to see how it works. You can have different devices, but the more common is a kind of syringe that has the fiber in a plug, and then you can work in two different works. You can introduce the fiber in the sample, wait until the analytes are absorbed, and then thermally dissolve in a GC. Or you can also dissolve in an HPLC interface, or you can use the headspace solid phase microstructure, and this is a method that is used for analytes that are slightly volatile, and it has as an advantage that not only you can concentrate a very, very high volume of sample, but you can also retain many, many analytes. Then you have all the compounds. Here you have other devices, like for example, the in-tube solid phase microstructure. The difference between the fiber and the in-tube is that in the case of the in-tube, the sample passes through a hollow capillary, and then the inner part of the capillary is that retains the analytes. You have here the stir bar sortive extraction, where you can see different different device in which the stir bar is slightly higher than the fiber, and then is able to retain higher amount of analytes. And you see that also the stir bars can be used with liquid chromatography. And then the other aspects of microstructure is to use just a drop of a solvent, of an immiscible solvent, to be introduced in water and to absorb the analytes. Commonly, these techniques are very, very delicate, and then one alternative to these techniques is the use of hollow fiber membranes, and then the pass of the analytes through them. This helps to retain some of the compounds, and when you have here how single drop microstructure works, in the case of the single drops, the difficult part of this is to keep the drop in the drop in the medium, but you can use it immersed in water in the head space. You can use, you can play with several layers of donor phases, acceptor phases, etc. And finally, you have again the membrane assist liquid microstructure that makes the technique more robust, because you could imagine that to keep a drop of organic solvent in a medium is complicated. And finally, the most used technique, that is the dispersive liquid microstructure, in which demonstrate how simplicity gains other determinations, because you have here how to do it. Commonly use a disperser solvent with water that could be acetone and nitrate, and some immiscible solvent that could be dichloromethane, hexane, ethyl acetate, and then you have here a couple of examples showing how it works with solvent that are denser than water and solvent that are less dense than water. With this, you have seen more a rapid overview in this tutorial of how compounds could be distinguished, could be isolated and extract for water. Then, I don't know if you have any question. If you have, we can enlarge this and answer the questions. And then, otherwise, thank you very much for your attention.
Thank you, Yolanda, for an exhaustive talk on this sample preparation, sampling procedures, and that's a beautiful compilation of all the methods, all the techniques which we use for chromatographic or for any environment technique for environmental analysis. So participants, if you have any query related to this talk or in general any query related to this course, please raise your hand, and Yolanda is here, local coordinator, Dr. Malik is here. There is no query from any participant.
Indu, Harpreet, Kshitij, Akshitij, Kshitij, Madhura, Neha, Poonam, Renuka, Seema, Simrat, Vaheguru, Sahil. Hello, Sahil.
There is a point, even if you don't have any query, and if coordinator is asking your name, at least you could respond. Just by saying, sir, I don't have any query.
Nothing, they don't have questions. Well, in this case, I think that this is the last class of the course, no? Then, well, thank you very much and see you tomorrow.
Yes, Yolanda.
Okay, very nice to see you. Bye-bye. Well, in any case, if someone have a question, you have my email and you can write me or anything, okay? Bye.
Bye-bye.
Have a nice weekend and evening.
The same to you. Have also a nice weekend. Well, the same to everybody. Nice weekend. See you tomorrow.
Yes.
Our weekend is going to be with GIAN only. And that will be a great weekend. Because
Okay.
we will be learning in this weekend also.
Okay. Yes, because the course is also for the weekend, but
Yeah.
well, thank you very much.
Thank you. Thank you so much.